When we think about the human body, we often picture the whole form, the shape of the face, the steady rhythm of breathing, or the quiet beating of the heart. Yet beneath these familiar images lies a hidden world. It is a world too small for our eyes to see unaided, but it is the foundation of everything we are. This hidden world is the realm of cells. Every sensation, every movement, every quiet thought depends on these microscopic structures, though they number in the trillions, and though each one is too small to notice on its own. Together they form the orchestra of life. They work silently, without pause, keeping balance within us as we go about our days. Even as you listen now, countless cells within you are exchanging energy, sending signals, and renewing themselves. Cells are the building blocks of life. They are not lifeless particles, but tiny living units, each with its own boundaries, its own tasks, and its own rhythms. They gather nutrients from the world around them, transform them into usable energy, and dispose of waste with remarkable efficiency. In essence, a cell is like a small community, contained within delicate walls. Within it, countless molecules are arranged into systems, each contributing to the overall harmony. It is a curious thought. Everything you have ever done, from the first breath you took as an infant to the movements of your muscles as you lie down to rest, has been supported by cells. They repair themselves when damaged. They multiply to replace what has been lost. And they communicate in subtle ways to ensure that the larger whole, the human being, remains in balance. To imagine a cell, it helps to picture a tiny sphere of water enclosed by a fine membrane. Inside that droplet lies a complex landscape. There are structures that act like power stations, others that resemble workshops, and still others that serve as storage or information libraries. Each part has its own role, and yet none could function without the others. A cell is at once simple in appearance and intricate beyond measure. Though they are small, cells are not all the same. Some are round and smooth, like red blood cells that drift through the bloodstream, delivering oxygen. Others are long and thread-like, such as nerve cells that stretch across the body, transmitting messages at remarkable speed. Muscle cells are packed with fibers that contract and release, producing the movements we so often take for granted. Skin cells form protective layers, renewing themselves in a continuous cycle. Each type of cell, in its own way, is tailored to a specific task, and together they form the tissues and organs that make life possible. For most of human history, this microscopic world was unknown. People understood the body through what they could see and feel. Muscles could be flexed, bones could be broken, and blood could be seen flowing, but the invisible world beneath remained a mystery. It was only with the invention of the microscope in the 17th century that humanity first glimpsed the cellular foundations of life. In those early days, what was seen appeared almost like a honeycomb, tiny compartments that seemed simple and empty. Yet over time, as instruments improved, those compartments revealed themselves to be bustling with unseen activity. Today, when scientists peer through advanced microscopes, they observe a landscape that is at once orderly and dynamic. Molecules move in steady flows, organelles shift and divide, and signals ripple across the cell's surface like gentle currents in water. Each movement has meaning. Each motion is part of a pattern that maintains life. As we begin this quiet journey through the basics of human cells, it may be helpful to think of the body as a vast city. The organs are like districts, each with its own purpose, and within those districts are countless houses. The houses themselves are the cells. Within them, life carries on in miniature. Some houses light up brightly with energy, others are filled with information libraries, while others work as factories, producing the materials that the rest of the body needs. And just as cities rely on roads and communication systems to function, cells too are linked, exchanging signals and resources to ensure that the greater whole remains stable. This invisible city of cells is not silent, even if we cannot hear it. Proteins fold and unfold, enzymes spark chemical reactions, and genetic material is carefully copied and passed along. All of this happens without us noticing. It is a background rhythm, steady and patient, like waves against a shore. In this way, the study of cells is not just a scientific pursuit, but also a gentle reminder of life's quiet complexity. Beneath the surface of every living being lies this miniature universe, intricate yet harmonious, busy yet balanced. To understand it, even in simple terms, is to gain a new appreciation for the hidden depths of the human body. As we move deeper into this session, we will explore what a cell is made of, how it came to be understood, and the many ways it sustains us. For now, it is enough to hold the thought that inside you, even in this very moment, a world of cells is at work. They do not rest, they do not falter, and they do not ask for recognition. They simply carry on, allowing you to breathe, to listen, and, perhaps, to drift quietly into rest. 2. What is a cell? A cell, at its simplest, is the most basic unit of life. It is the smallest structure that we can truly call alive, and yet it carries within it all the machinery necessary to sustain existence. If something is alive, whether it is a floating bacterium, a spreading oak tree, or a human being, it is made of cells. This is one of the most fundamental truths of biology, a truth that quietly ties together all of life on Earth. To understand the idea of a cell, we might imagine life as a great woven fabric. Each thread in that fabric represents a single cell, and when billions or even trillions of those threads are brought together, the larger design appears. For humans, the design is the body, our tissues, our organs, and all the functions that keep us breathing and moving. And yet, even if you were to take a single thread from this fabric, a lone cell, you would still find life's essence contained within it. A single cell, on its own, is able to perform many of the same processes as the complete organism. It takes in food, it transforms energy, it eliminates waste, it grows, and under the right conditions, it reproduces. It is easy to overlook this, because cells are far too small for the naked eye to see. They measure in micrometers, a scale so fine that hundreds of them could fit across the width of a human hair. Despite their smallness, they are not lifeless particles of dust. They are living compartments, organized and purposeful, each one capable of maintaining its own identity, while also contributing to the greater whole. At the heart of the cell's structure is a delicate boundary called the plasma membrane. This membrane is like a soft, flexible skin. It is not simply a barrier, but a careful gatekeeper. The membrane decides what may enter and what must be kept out. Nutrients and oxygen are welcomed in, while harmful substances are blocked. Waste products, which if left inside would cause harm, are gently passed out. This ability to regulate exchanges is essential because it allows the cell to maintain a stable internal environment, even when conditions outside may be changing. Inside this membrane lies the cytoplasm, a fluid interior that holds all the working parts of the cell. Within this soft medium float organelles, the tiny specialized structures that carry out different tasks. The word organelle means little organ. And just as our body has organs that serve unique purposes, the cell too has these miniature organs. Some organelles are devoted to producing energy. Others build proteins. Some store important materials. And some carry the instructions of life itself. To picture a cell, one might think of a self-contained workshop. The outer wall of the workshop is the plasma membrane, keeping everything inside safe and orderly. The workers inside are the organelles, each carrying out a particular duty. Some workers focus on breaking down raw materials into usable parts. Others carefully build proteins, folding them into just the right shapes. Some workers are responsible for storage, making sure nothing essential is lost. And others hold the blueprints, the genetic code that guides every step of the process. This workshop is not static. It is always active, always adjusting, always finding balance. Molecules move in steady streams, signals are exchanged, and new products are made, moment by moment. All of this happens without us noticing, yet without it we could not exist. The diversity of life begins at this microscopic level. There are organisms, such as bacteria, that consist of a single cell. These solitary beings manage everything within their tiny boundaries, living entire lives as self-sufficient units. They remind us that the cell is not only a part of life, it can be life itself. At the other end of the spectrum are multicellular organisms, such as plants, animals, and of course humans. In these bodies, trillions of cells work together in harmony. No one cell carries the burden alone. Instead, they specialize. Some become muscle cells, capable of contracting and producing movement. Others become nerve cells, long and delicate, carrying messages across the body at remarkable speed. Still others become blood cells, round and flexible, designed to travel and deliver oxygen. Skin cells form protective layers, constantly renewing themselves to shield the body from harm. What is remarkable is that despite their differences, all of these cells share the same basic plan. They all are enclosed by a membrane. They all contain genetic material. They all transform energy to keep themselves alive. They all have the ability to grow and divide. These common features reveal a unifying truth, that all living things, no matter how different they appear on the surface, are tied together by the cellular foundation of life. It is humbling to reflect on how complex even the smallest cell truly is. Within the span of a single second, thousands of chemical reactions take place inside. Nutrients are broken down into smaller pieces. Proteins are constructed and folded. Waste is identified and expelled. And energy is transformed into usable forms. Each reaction is carefully timed, and each depends on countless molecules working in harmony. The precision of it all is astonishing, and yet it is not something we ever consciously notice. It simply continues, quietly and dependably, within us. There is also beauty in the way structure and function are linked at this level. The shape of a cell often hints at what it does. The long, thread-like form of a neuron reveals its purpose: to connect distant parts of the body, like wires carrying signals. The flattened layers of skin cells show their role in covering and protecting. The flexible roundness of red blood cells allows them to squeeze through narrow vessels, reaching every corner of the body with their life-giving oxygen. In this way, the design of each cell is intimately connected to its duty, a reflection of the larger principle that form and function are inseparable. Even though cells can be studied in isolation, they rarely exist that way in our bodies. Instead, they gather together in groups called tissues. Similar cells unite to form tissues, and those tissues in turn combine into organs. Organs work together in systems, and those systems sustain the entire organism. This layered organization from cell to tissue, tissue to organ, organ to system, and finally, system to body, shows how life builds itself from the bottom upward, with the cell as the essential foundation. And yet, even while they contribute to something greater, each cell maintains its individuality. Each one keeps its own balance, repairs its own damage, and follows its own cycles of growth and division. This combination of independence and cooperation is what makes the living body possible. It is perhaps worth pausing to consider the quiet resilience of cells. They do not seek recognition. They work continually, repairing themselves when injured, multiplying to replace what has been lost, and carrying out their roles even as we sleep. They are the unseen guardians of life, small yet powerful, fragile yet enduring. To study cells is to step into a world of hidden detail. It is to realize that beneath every thought, every movement, and every breath lies a foundation built of countless invisible parts. Cells are both the beginning and the sustaining force of life. They are not visible to the eye, and yet they are everywhere, composing the body and keeping it alive moment by moment. In the next section, we will step back into history and follow the story of how cells were first discovered. Before microscopes revealed their hidden presence, people had no way of knowing that life's essence was made of these tiny structures. The tale of their discovery is a story of curiosity and patience, of lenses polished with care, and of the first glimpses into a world that would forever change our understanding of life. 3. The discovery of cells. For most of human history, the cell remained invisible. People lived, worked, and wondered about the human body, without ever suspecting that it was made up of countless tiny units. Illnesses were explained in terms of humors, energies, or spiritual influences. The body itself was seen as a single, continuous structure, not a collection of microscopic parts. It was not until the invention of the microscope in the 17th century that this hidden world came into view, and with it, a quiet revolution began in biology. The first glimpses of cells came through the work of Robert Hooke, an English scientist and philosopher. In 1665, Hooke published a book titled Micrographia. Within its pages were detailed drawings of objects he had examined through a compound microscope, a device made of two lenses aligned to magnify tiny details. One of his subjects was a thin slice of cork. Under the magnifying lenses, Hooke noticed a pattern of small, box-like compartments. They reminded him of the simple rooms, or cells, in a monastery, and so he gave them that name. The term has remained ever since. What Hooke saw, however, were not living cells as we understand them today. Cork is the dead bark of a tree, and the compartments Hooke observed were empty cell walls, lifeless chambers that once housed living material. Still, the image was striking enough to capture the imagination of his readers. For the first time, the concept of life being built from small, repeating units began to take shape. Soon after, the Dutch tradesman and lensmaker Antony van Leeuwenhoek pushed the story further. Unlike Hooke, Leeuwenhoek did not use a compound microscope, but instead crafted exquisitely polished single lenses. With these simple but powerful instruments, he achieved magnifications greater than those of Hooke's microscope. Around 1674, peering into drops of pond water, Leeuwenhoek described tiny moving creatures he called animalcules. These were, in fact, single-celled organisms, such as protozoa and bacteria. Leeuwenhoek's observations were extraordinary. He reported seeing red blood cells coursing through vessels, sperm cells swimming with delicate motion, and microscopic fibers of muscle. His letters to the Royal Society in London, though sometimes greeted with skepticism, gradually convinced the scientific world that life extended into realms too small to be seen with the naked eye. With Leeuwenhoek's discoveries, cells were no longer imagined as static compartments, as Hooke had drawn them, but as dynamic, living entities full of motion and purpose. Through the 18th century, microscopes slowly improved, and naturalists continued to gather observations. Still, it was not until the 19th century that a coherent theory of cells began to form. Two German scientists, Matthias Schleiden, a botanist, and Theodor Schwann, a zoologist, each studying plant and animal tissues respectively, reached a unifying conclusion. In the 1830s, they declared that all living organisms, whether plant or animal, are made of cells. This statement formed the foundation of what is now known as the cell theory. The theory grew further when Rudolf Virchow, another German scientist, added an essential insight in 1855. He declared, "Omnis cellula e cellula." All cells arise from pre-existing cells. This was a turning point. It meant that life was not continuously generated anew from non-living matter, as some once believed, but instead carried forward through the division of cells into more cells. Each cell was a continuation of life from one generation to the next, part of an unbroken chain stretching back through time. At this point, the pieces had come together. All living things are made of cells. The cell is the basic unit of structure and function, and new cells come only from existing ones. These three principles form the modern cell theory, still one of the cornerstones of biology. It is interesting to reflect on how limited the early instruments were, and yet how much they revealed. Hooke's compound microscope, by today's standards, was crude. Its lenses distorted light, creating blurred or colored edges. Leeuwenhoek's lenses, though clearer, offered only a small field of view. And yet, with patience and careful observation, these pioneers uncovered truths that transformed our understanding of life. As the 19th and 20th centuries unfolded, microscopes became more powerful and refined. The invention of the light microscope with improved optics allowed scientists to see more detail, such as the nucleus inside the cell. Later, in the 20th century, the development of the electron microscope opened entirely new horizons. Instead of using light, electron microscopes used beams of electrons, achieving magnifications thousands of times greater. For the first time, scientists could see the intricate structures of organelles, the folded membranes of mitochondria, the stacked discs of the Golgi apparatus, the delicate scaffolding of the cytoskeleton. The cell was no longer a blurred shape, but a detailed, bustling landscape. Beyond microscopes, new methods such as staining techniques, fluorescent labeling, and live imaging brought the cell to life in vibrant detail. Researchers could watch molecules moving, proteins folding, and entire networks of communication unfolding within and between cells. Each advance deepened the sense of wonder, showing that the smallest units of life were more complex than anyone had imagined. And yet, even with the most sophisticated technology, the spirit of those early discoveries remains. Hooke's careful drawings, Leeuwenhoek's patient observations, Schleiden and Schwann's unifying vision, all of these moments were quiet steps in unveiling the hidden architecture of life. They remind us that great revolutions in knowledge often begin not with loud declarations, but with quiet attention to detail. Today, when we speak of cells, we do so with the benefit of centuries of progress. We know that our bodies are made of trillions of them. We know that inside each cell is a library of DNA, a record of instructions reaching back to our earliest ancestors. We know that cells communicate, cooperate, and adapt, forming tissues and organs that carry out life's functions. But it is worth remembering that this knowledge began with simple lenses, steady hands, and curious minds. The discovery of cells changed not only biology, but also our place in the universe. It revealed that life is built on a repeating pattern, that the smallest structures carry within them the power of growth and continuity. It showed us that the human body, vast and complex as it seems, is composed of tiny units, each alive in its own right. As you rest and reflect, it is perhaps calming to think of the quiet history behind our understanding. The discovery of cells is not a tale of sudden revelation, but of gradual unveiling. It is the story of patient observation, of glass lenses polished with care, of scientists peering into drops of water or thin slices of cork, and finding within them the building blocks of life. It is a story that begins in silence, unfolds slowly, and continues even now, as microscopes grow sharper and knowledge deepens. And so, from unseen chambers in cork to the bustling interiors revealed by modern imaging, the discovery of cells has been a journey of looking closer and closer. Each step has brought us nearer to the realization that beneath the visible world lies another, smaller one, filled with life's quiet activity. In the next part, we will explore the cell theory in greater depth. We will return to those three principal cells as the units of life, the basic structures of organisms, and the continuity of cells through division, and see how they form the framework of modern biology. For now, let the thought settle gently. Every living being, every plant, every animal, and every human is made of cells, a truth uncovered slowly through centuries of quiet discovery. 4. The cell theory. When we look back on the gradual discoveries that revealed the existence of cells, what emerges is not just a collection of observations, but a framework, a way of understanding life itself. This framework became known as the cell theory. It is built upon three simple but powerful principles. All living things are made of cells. The cell is the basic unit of structure and function in every organism, and every new cell arises from an existing cell. These ideas may seem almost obvious to us now, so deeply woven into our knowledge that we rarely stop to question them. But when they first appeared, they represented a profound shift in how people saw life. Before the cell theory took shape, it was widely believed that living things could simply appear from non-living matter. The air might give rise to flies, damp soil might generate plants, stagnant water might spawn tiny creatures. This view, called spontaneous generation, lingered for centuries. It was difficult for people to imagine that life could be built from units too small to see, or that the continuity of life might be carried quietly from one generation to the next. through invisible processes. The cell theory overturned these ideas. It revealed that life had a deeper order, a structure that ran through every plant, every animal, and every human being. The first principle of the theory is that all living organisms are composed of cells. For the smallest bacteria, life is contained within just one. That single cell performs every task required to survive. It takes in nutrients, transforms energy, responds to its environment, and divides to reproduce. For more complex organisms, such as plants and animals, the story is more elaborate. These beings are formed from trillions of cells, each one specialized for a different role. Muscle cells contract, skin cells form barriers, nerve cells carry signals, and blood cells drift like couriers through the bloodstream. Yet, however different their duties, they all share the same basic identity. They are cells. This principle is unifying. It tells us that every living creature on Earth, no matter how varied in form or behavior, is built from the same fundamental units. The second principle states that the cell is the basic unit of structure and function. This means that the cell is the smallest living structure, capable of carrying out all the processes of life. Below this level, matter is chemical. Molecules, atoms, and minerals may form the ingredients of life, but they are not alive. Above this level, tissues and organs exist, but only because they are made from cells. The cell is the threshold where non-living matter becomes living. Within its delicate boundaries, energy is harnessed, proteins are built, waste is expelled, and instructions are stored. It is like a small, self-contained world. Inside its thin membrane is a kind of workshop where every essential task is performed. Some parts of the workshop act like kitchens, generating energy. Others serve as storerooms, keeping important supplies safe. There are workbenches where proteins are carefully folded, and a central library where the genetic script of life is kept. Despite its size, the cell is complete. Nothing smaller can sustain life as we know it. The third principle tells us that all cells come from pre-existing cells. This statement emphasizes continuity. Life does not simply appear out of nowhere. Each new cell is born from the division of another. A parent cell copies its instructions, divides its contents, and passes them on to two daughter cells. This is how growth occurs, how tissues renew themselves, and how life continues from one generation to the next. The idea seems simple, but when it was first expressed, it was revolutionary. It shattered the belief in spontaneous generation and replaced it with a vision of life as an unbroken chain. Every cell in the human body today is descended from other cells, and those from still earlier ones, stretching back in a line that reaches into the earliest history of life on Earth. Though the three principles are straightforward, their implications are vast. They explain why the body grows, why wounds heal, why diseases spread, and why life is continuous. They underpin medicine, genetics, and molecular biology. They remind us that beneath the complexity of tissues and organs lies a common pattern. Every heartbeat, every breath, every thought is sustained by countless cells following these same quiet rules. Over time, the cell theory has been gently expanded. We now understand that the health of an organism depends on the health of its cells, that the activity of tissues and organs reflects the sum of the activity within individual cells. We also know that within every cell lies the genetic material of life, encoded in DNA, and that this information is faithfully passed from one cell to another. We recognize that cells are not static but constantly transforming energy, drawing in nutrients, breaking them down, and fueling their own processes. This flow of energy sustains the balance of life within each cell and within the body as a whole. It is calming to consider how much order exists within this framework. Billions of cells are working within you now, each one balancing, dividing, and repairing itself. And yet they are not acting in chaos. They are following patterns that have endured for billions of years, repeating the same processes that sustained the first living cells. The cell theory reveals life as something both intricate and orderly, both diverse and unified. It is a reminder that, beneath the surface, everything alive is built from the same repeating units. There is also something deeply reassuring in the thought that the cells within you are part of an unbroken chain. Each one carries the continuity of life forward, a link in a chain that has never been broken since the first cells appeared on Earth. They are quiet witnesses to time, passing on the instructions of life from one generation to the next. They do this without recognition, without pause, sustaining existence in the most patient way imaginable. As you rest with this thought, it is enough to hold on to the three principles. All living things are made of cells. The cell is the basic unit of life, and every new cell comes from an existing one. These are the foundations upon which modern biology is built. They are also gentle reminders of life's hidden unity, of the quiet architecture beneath every leaf, every creature, and every human body. In the next part, we will turn to the distinction between the simplest forms of cells and the more complex ones. We will see how prokaryotic cells, small and ancient, differ from eukaryotic cells, large and intricate. By exploring this contrast, we can better appreciate the variety of life at its smallest scale and the ways in which these differences echo throughout the living world. 5. Prokaryotic cells. When we consider the wide variety of cells that exist, it is useful to begin with the simplest forms, for in their simplicity lies both age and endurance. These are the prokaryotic cells, a name that comes from Greek roots meaning before nucleus. Prokaryotes are among the oldest living beings on Earth, thought to have appeared more than 3.5 billion years ago, long before plants, animals, or even complex single-celled organisms evolved. They are small, usually only a few micrometers across, and they lack the intricate compartments that we find in more advanced cells. And yet, despite their modest design, they have survived and thrived in nearly every environment the planet has to offer. The hallmark of prokaryotic cells is their simplicity. They have no nucleus, no elaborate system of organelles, no complex inner compartments. Their genetic material is not enclosed in a protective structure, but instead floats within the cell in a region called the nucleoid. This DNA is typically circular, a long thread carrying the instructions that allow the cell to grow, divide, and adapt. Around this core lies the cytoplasm, a watery interior where enzymes and molecules move about, carrying out the chemical reactions that sustain life. The entire cell is wrapped in a plasma membrane, a delicate yet essential barrier that regulates what enters and what leaves. Outside of this, most prokaryotes possess a rigid cell wall, a protective shell that gives them shape and strength. Some also produce an additional layer called a capsule, a slimy coating that helps them cling to surfaces and evade threats. These layers make prokaryotes remarkably resilient. They can survive in soil, in water, in the air, and even in extreme environments where other forms of life would perish. Despite their lack of complexity, prokaryotic cells are far from powerless. Within their tiny space, countless reactions unfold every second. Enzymes break down food molecules, energy is harvested, and new proteins are built. Many prokaryotes move about, propelled by whip-like structures called flagella, or tiny hair-like appendages called pili. These structures allow them to swim through water or crawl across surfaces, seeking food or new places to live. There are two main groups of prokaryotes, the bacteria and the archaea. Bacteria are perhaps the more familiar, for they are everywhere. They live in the soil, where they recycle nutrients and enrich the earth. They live in water, where they form part of the foundation of aquatic food chains. They live inside the human body, in the mouth, on the skin, and most abundantly in the intestines, where they help digest food and maintain balance. While some bacteria can cause illness, many more are harmless or even beneficial, reminding us that life is interconnected at every level. The archaea, though less well known, are equally fascinating. Once thought to be simply unusual bacteria, they have since been recognized as a distinct domain of life, with unique genetics and biochemistry. Archaea are often found in extreme environments. Some live in scalding hot springs, others in salty lakes, and others still in oxygen-free swamps. They thrive in places that seem inhospitable, showing us that life can adapt to conditions once thought impossible. Their resilience even raises questions about whether life might exist in similar extreme environments elsewhere in the universe. The simplicity of prokaryotic cells also makes them efficient. Because they lack the internal divisions of more advanced cells, their processes unfold directly within the cytoplasm. DNA is transcribed into RNA, which is immediately translated into proteins by ribosomes floating nearby. Growth and reproduction occur quickly. Many prokaryotes reproduce by binary fission, a process in which the cell copies its DNA, elongates, and then splits in two. Under favorable conditions, this can happen astonishingly fast, sometimes in less than half an hour. A single bacterial cell could give rise to millions of descendants in a single day, a reminder of how prolific and adaptable these tiny beings can be. Yet even in their simplicity, prokaryotic cells display remarkable strategies. Many can exchange small fragments of DNA with one another, passing along useful traits such as resistance to certain chemicals or the ability to metabolize new foods. This sharing of genetic information allows them to adapt swiftly to challenges, ensuring their survival in changing environments. It is a quiet form of communication, one cell passing instructions to another, strengthening the community as a whole. If we step back and consider the role of prokaryotic cells in the history of life, their significance becomes clear. For billions of years, they were the only life forms on Earth. During this time, they shaped the atmosphere, created new biochemical pathways, and laid the foundation for more complex organisms to evolve. It was prokaryotes that first learned to capture energy from sunlight, releasing oxygen into the air and transforming the planet's environment. Without their ancient activity, the world would not be as it is today, and multicellular life would never have arisen. Even now, they continue to sustain the balance of ecosystems. In the oceans, photosynthetic bacteria form the base of the food web, producing energy that supports countless other creatures. In soils, decomposing bacteria break down dead material, recycling nutrients that plants can use. Within our own bodies, communities of bacteria help digest food, synthesize vitamins, and train our immune systems. They are everywhere, quietly active, often unnoticed, but always essential. The small size of prokaryotic cells belies their importance. They remind us that complexity is not always necessary for survival. In their minimal design, they carry all that is needed for life, and they carry it well. They have endured for billions of years, adapting to every challenge, inhabiting every niche. While we often think of ourselves and other large organisms as the dominant forms of life, it is in fact the prokaryotes that have persisted the longest and remain the most numerous. They are the unseen majority, shaping the world in ways both visible and invisible. As you reflect on this, it may be calming to remember that the story of life is not only one of grand structures and complex organisms. It is also a story of simplicity, of tiny cells carrying on quietly, repeating the same ancient processes that sustained the first living beings. Prokaryotic cells embody this continuity. They are the first chapter of life's history, and even now, they remain its most enduring presence. In the next part, we will turn to the more complex cousins of the prokaryotes, the eukaryotic cells, larger and more elaborate, filled with specialized compartments. These cells form the foundation of plants, animals, fungi, and many other organisms. By exploring their structure, we can begin to see how complexity arose from simplicity and how life diversified into the forms we know today. 6. Eukaryotic cells. If prokaryotic cells represent life in its most ancient and simple form, then eukaryotic cells represent the next great step in biological complexity. The word eukaryote comes from Greek roots meaning true nucleus, and that is the defining feature of these cells. Their genetic material is enclosed within a membrane-bound compartment called the nucleus. This seemingly small difference has profound consequences. It allows eukaryotic cells to separate the delicate instructions of DNA from the rest of the cellular environment, creating space for greater control, regulation, and specialization. From this foundation has arisen the vast diversity of plants, animals, fungi, and many single-celled organisms known as protists. Eukaryotic cells are larger than prokaryotic cells, often 10 times the size or more. With this increase in size comes an increase in internal complexity. Rather than containing all processes within one shared space, eukaryotic cells divide themselves into compartments, each with its own membrane and its own tasks. These compartments are known as organelles, and they function much like the organs of a larger body. Within them, energy is produced, proteins are folded and modified, materials are packaged and shipped, and waste is broken down. The result is a level of organization that allows eukaryotic cells to perform a wide range of functions, supporting the complexity of multicellular organisms. At the heart of the eukaryotic cell lies the nucleus, the repository of genetic information. Within its double membrane are long strands of DNA, coiled and folded into chromosomes. The nucleus is not merely a storage site, but also a place where DNA is copied into RNA, which then carries instructions outward to guide the building of proteins. Around the nucleus lies the nucleolus, a dense region devoted to constructing ribosomes, the tiny machines that assemble proteins. By separating the nucleus from the rest of the cell, eukaryotes can carefully control when and how genes are expressed, allowing them to adapt with greater nuance to their surroundings. Outside the nucleus, the cytoplasm is filled with organelles, each performing specialized duties. The mitochondria, often called the powerhouses of the cell, transform nutrients into usable energy. Through a process known as cellular respiration, they take in molecules of glucose and oxygen and convert them into adenosine triphosphate, or ATP, the energy currency that drives nearly every cellular activity. The importance of mitochondria is difficult to overstate. They are the reason muscles can contract, the reason neurons can fire, the reason cells can divide. Their constant work sustains the flow of energy that allows the body to live. Surrounding the nucleus are networks of folded membranes that form the endoplasmic reticulum. One region, known as the rough endoplasmic reticulum, is studded with ribosomes that build proteins. These proteins enter the folds of the membrane, where they are shaped, modified, and prepared for their roles. Another region, the smooth endoplasmic reticulum, carries out different tasks, such as building lipids and detoxifying harmful substances. From there, materials are sent to the Golgi apparatus, a series of flattened sacs that act like a post office of the cell, packaging proteins and lipids into small vesicles and sending them to their destinations within the cell or to the outside world. Other organelles contribute in quieter ways. Lysosomes contain digestive enzymes that break down waste and recycle worn-out components, ensuring that the cell remains clean and efficient. Peroxisomes carry out chemical reactions that neutralize toxins and break down fats. The cytoskeleton, a network of protein filaments, provides shape and structure, while also serving as tracks along which vesicles and organelles can move. Tiny molecular motors walk along these tracks, carrying cargo to where it is needed, a constant flow of traffic in the bustling city of the cell. In plant cells, eukaryotic complexity is even more evident. Along with the nucleus, mitochondria, and endoplasmic reticulum, they contain chloroplasts, organelles that capture energy from sunlight and convert it into food through photosynthesis. Within their green membranes, carbon dioxide and water are transformed into glucose, releasing oxygen as a byproduct. These chloroplasts are the reason plants can grow toward the light, and they are the foundation of nearly all food chains on Earth. Without them, life as we know it would not be possible. One of the most intriguing aspects of mitochondria and chloroplasts is their origin. Scientists believe that billions of years ago, ancient prokaryotic cells entered into a partnership with larger host cells. Instead of being digested, they remained inside, providing energy or photosynthetic abilities in exchange for protection and nutrients. Over time, they became permanent residents, evolving into the mitochondria and chloroplasts we see today. This theory, known as endosymbiosis, reminds us that even the complexity of eukaryotic cells has roots in cooperation and adaptation. It also shows that the history of life is not simply one of competition, but also of quiet partnerships that endure across the ages. Eukaryotic cells, with their ability to divide labor among organelles, pave the way for multicellularity. When individual cells join together, some specialized in movement, others in sensing the environment, others in providing structure. Gradually, these cooperative groups became tissues and organs, forming the bodies of animals and plants. Without the complexity of eukaryotic cells, such specialization would not have been possible. It is their flexibility and capacity for organization that allow the human brain to think, the heart to beat, and the forest to grow. Though complex, eukaryotic cells are not invulnerable. They must maintain balance, repairing damage, managing energy, and responding to signals from their environment. If this balance falters, the consequences ripple outward, sometimes resulting in disease. Yet in their quiet way, most eukaryotic cells manage these challenges with remarkable stability, carrying out their tasks day after day, often for years. Some cells in the human body divide rapidly, renewing themselves often, while others, such as certain neurons, may last for a lifetime. As we reflect on these cells, it is helpful to imagine them not as mechanical objects, but as small communities, each with its own members and responsibilities. They are intricate yet orderly, each organelle performing its task in harmony with the rest. Together, they embody the idea that complexity can arise from simplicity, that from small beginnings can come great diversity. The contrast between prokaryotic and eukaryotic cells is striking, yet both are bound by the same fundamental principles. Both rely on membranes to create boundaries. Both carry genetic instructions in DNA. Both transform energy to stay alive. The difference lies in how these tasks are arranged and carried out. In the simplicity of prokaryotes, we see endurance and efficiency. In the complexity of eukaryotes, we see specialization and diversity. Both paths have been successful, coexisting side by side for billions of years, shaping the living world together. As you settle with these thoughts, it is worth holding on to the image of the eukaryotic cell as a small but complete world, filled with activity, yet balanced in its order. It is the foundation of every tree, every flower, every animal, and every human being. It carries within it the echoes of ancient partnerships and the promise of endless variety. Quietly, patiently, it sustains life. In the next part, we will focus on one of the most important features shared by all cells, simple or complex, the boundary that encloses them, the cell membrane. By exploring this delicate yet powerful structure, we can see how the cell maintains its integrity, communicates with its surroundings, and regulates the steady flow of life within. 7. The cell membrane. At the edge of every cell lies a boundary so thin that it cannot be seen without powerful instruments, and yet so essential that life would not be possible without it. This is the cell membrane, also known as the plasma membrane. It is the delicate skin of the cell, the line that separates inside from outside, self from environment. Though only a few nanometers thick, it is one of the most important structures in biology, for it maintains order, balance, and communication. The cell membrane is often described as selectively permeable, meaning that it allows some things to pass through while keeping others out. This selectivity is the reason a cell can maintain its own internal environment, distinct from the world around it. Without such control, the cell would be flooded with harmful substances or drained of vital components. Instead, the membrane stands guard, admitting nutrients, letting out waste, and permitting only certain signals to enter. In this way, the cell membrane acts like both a shield and a gateway, protecting the cell while also connecting it to its surroundings. The structure of the membrane is a marvel of simplicity and elegance. It is built primarily from molecules called phospholipids, which arrange themselves in a double layer, known as the phospholipid bilayer. Each phospholipid has a head that is drawn to water and a tail that avoids it. When placed in an aqueous environment, these molecules spontaneously line up, with the water-loving heads facing outward toward the fluid inside and outside the cell, and the water-fearing tails tucked safely inward, away from the water. The result is a self-assembling sheet, flexible yet stable, that forms the basic fabric of the membrane. But the membrane is not made of lipids alone. Embedded within this bilayer are proteins of many kinds, each carrying out specific duties. Some proteins act as channels, forming passageways through which ions and small molecules can move. Others act as pumps, using energy to transport substances against their natural flow. Still others serve as receptors, detecting signals from the environment or from neighboring cells, and transmitting those messages inward. These proteins give the membrane much of its function, transforming it from a simple barrier into a dynamic interface. One of the remarkable qualities of the cell membrane is its fluidity. The molecules within it are not locked in place but drift and shift like boats on water. Proteins move laterally, lipids exchange positions, and the entire structure remains flexible. This fluid mosaic, as it is often called, allows the membrane to adapt to changes, to engulf materials in processes such as endocytosis, or to release substances through exocytosis. It is not a rigid wall but a living boundary, constantly in motion, constantly reshaping itself to meet the needs of the cell. The cell membrane also plays a crucial role in communication. On its outer surface are carbohydrates attached to proteins and lipids, forming patterns much like identification tags. These molecular tags help cells recognize one another, distinguishing self from non-self, friend from invader. In the immune system, this recognition is vital, allowing defense cells to detect harmful intruders while leaving the body's own cells unharmed. In tissues, it allows cells to adhere to one another, forming the structures that hold organs together. Through these subtle signals, the membrane becomes a language of contact and cooperation, beyond protection and communication. The cell membrane is deeply involved in the regulation of energy and balance. It controls the flow of ions such as sodium, potassium, and calcium, maintaining gradients that are essential for processes like nerve transmission and muscle contraction. It balances the intake of nutrients with the release of waste, ensuring that the internal environment remains steady even when the external environment changes. This constancy, known as homeostasis, is the foundation of life. The cell membrane is the quiet guardian that allows it to continue. If we imagine the cell as a small world, then the membrane is its atmosphere, its border, and its interface with the greater universe. It is not an impenetrable barrier but a permeable, responsive boundary, one that ensures safety while allowing connection. The balance it strikes between openness and protection is delicate, and yet it has endured for billions of years, supporting life in forms as varied as bacteria, trees, and human beings. The beauty of the cell membrane lies not only in its structure but also in its adaptability. Different cells modify their membranes in different ways to suit their needs. Neurons, for instance, fill their membranes with proteins that allow the rapid flow of ions, enabling the transmission of electrical impulses. Kidney cells shape their membranes into folded surfaces to maximize absorption. White blood cells extend their membranes outward to engulf invaders. Each adaptation reflects the specific role the cell plays within the body. And yet the underlying structure, the bilayer of lipids with embedded proteins, remains constant. It is humbling to realize that this thin layer, so easy to overlook, is the very reason life is possible. Without membranes, there could be no separation of inside and outside, no concentration of materials, no organization of reactions. The cell membrane is the condition that makes life's chemistry sustainable. It creates the space in which complexity can arise, protecting the fragile processes of life while still allowing the exchange of matter and energy with the outside world. As we pause with this thought, it becomes clear that the membrane is not merely a passive boundary but a dynamic, vital part of the cell's life. It is flexible, responsive, and deeply involved in every moment of cellular activity. It keeps the cell intact, it governs its exchanges, and it communicates its identity to the world. Though it is thin, it is strong. Though it is small, it is essential. In the next part, we will move beyond the boundary and into the inner compartments of the cell, exploring the organelles that make eukaryotic life possible. These specialized structures, each with its own duties, form the inner machinery of the cell. By understanding them, we can see how life organizes itself on the smallest scale, and how countless quiet processes combine to sustain the whole. 8. The organelles. Within the quiet interior of the cell lies a collection of specialized structures that together carry out the work of life. These are the organelles, a word that means little organs. Just as the body has organs dedicated to specific tasks, the heart to pump blood, the lungs to draw in air, and the stomach to digest food, so too does the cell contain its own set of compartments, each devoted to a particular role. They are suspended in the cytoplasm like islands in a sea, distinct yet interconnected, working in harmony to sustain the whole. The most central of these organelles is the nucleus, the keeper of genetic information. Enclosed within a double membrane, it protects the long strands of DNA that carry the instructions for building and maintaining the organism. Inside, the DNA coils around proteins, forming chromosomes, and within its dense regions, new instructions are constantly being transcribed into messenger molecules. These messenger RNAs carry the genetic code outward, guiding the construction of proteins in other parts of the cell. The nucleus also houses the nucleolus, a darker sphere where ribosomes are assembled before they travel out into the cytoplasm. The nucleus, with its protective shell and its constant transcription, is both archive and workshop, safeguarding the past while shaping the future. Floating in the cytoplasm are the ribosomes, tiny yet essential machines. They are not enclosed by membranes, but their function is vital. Ribosomes are the builders of proteins, reading the messenger RNA and assembling amino acids into chains. These chains fold into intricate shapes, becoming enzymes, structural proteins, or signaling molecules. Every movement, every reaction, every repair in the cell depends on proteins, and every protein begins at a ribosome. In this way, ribosomes are among the busiest and most fundamental of the organelles. Nearby lies the endoplasmic reticulum, a labyrinth of folded membranes stretching outward from the nucleus. One region, dotted with ribosomes, is known as the rough endoplasmic reticulum. Here, proteins are synthesized and modified, entering the folds where they are folded correctly and sometimes given chemical tags that guide them to their destinations. Another region, smooth in appearance, serves different roles, including the synthesis of lipids and the detoxification of harmful substances. This network, stretching like corridors through the cytoplasm, is a highway of construction and processing, ensuring that newly made molecules are prepared for use. From there, many proteins and lipids travel to the Golgi apparatus, a stack of flattened sacs that functions as the shipping center of the cell. Within these stacks, molecules are further modified, sorted, and packaged into small vesicles. Some are sent to the cell membrane to be released. Others are delivered to different parts of the cell. It is here that proteins gain their final touches, folded, trimmed, or tagged before they are sent out. The Golgi apparatus is both post office and finishing workshop, ensuring that every molecule reaches its correct destination. The mitochondria are another essential organelle, often called the powerhouses of the cell. They are oblong in shape, with folded inner membranes that create surfaces for chemical reactions. Within these folds, nutrients are broken down, and energy is captured in the form of ATP, the molecule that fuels nearly all cellular activity. Every thought, every heartbeat, every breath depends on this energy. What makes mitochondria especially fascinating is their origin. They are thought to have begun as independent prokaryotic cells that entered into a partnership with larger hosts billions of years ago. Instead of being digested, they offered energy. And over time, they became permanent residents, passing from one generation to the next. This ancient partnership endures in every eukaryotic cell today, a reminder of life's long history of cooperation. Plant cells contain another organelle born of ancient partnership, the chloroplast. These green structures capture the energy of sunlight, transforming carbon dioxide and water into glucose while releasing oxygen as a byproduct. Like mitochondria, chloroplasts contain their own DNA and ribosomes, reflecting their evolutionary past as once independent organisms. They are the reason plants grow toward the light, the reason forests rise and fields of grass spread, and they are the foundation of nearly all food chains, for the energy they capture sustains not only plants but also the countless animals that feed upon them. Other organelles carry out quieter but no less vital tasks. Lysosomes are small, spherical compartments filled with enzymes that break down waste and recycle components. They digest worn-out organelles, invading pathogens and excess materials, reducing them to simple molecules that can be reused. In this way, they are both custodians and recyclers, keeping the cell clean and efficient. Peroxisomes perform similar roles, neutralizing toxic substances and breaking down fats into usable forms. Their work is often unnoticed but indispensable, protecting the cell from harm. Beneath and throughout the cytoplasm stretches the cytoskeleton, a network of protein filaments that provide structure and support. These filaments act like scaffolding, giving the cell its shape, and also serve as tracks along which organelles and vesicles can move. Tiny motor proteins walk along these tracks, carrying packages from one part of the cell to another. The cytoskeleton also plays a role in cell division, helping separate chromosomes, and in movement, allowing some cells to change shape or migrate. Though invisible to the naked eye, it is a framework of strength and motion, always shifting, always maintaining balance. Taken together, these organelles transform the cell from a simple bubble of fluid into a complex, organized system. Each part has its task, and though they differ in structure and purpose, they work together in harmony. The nucleus provides instructions, ribosomes build proteins, the endoplasmic reticulum and Golgi apparatus prepare and ship them. Mitochondria and chloroplasts supply energy, lysosomes recycle, peroxisomes protect, and the cytoskeleton holds everything in place. The cell, with its organelles, becomes not just a unit of life but a community in miniature, a collection of tasks and workers sustained by balance and coordination. It is remarkable to consider how much activity unfolds within such a small space. Every second, molecules are moving, proteins are folding, energy is being captured, and signals are being sent. And yet, despite the apparent chaos, there is order. Each organelle plays its role, and together they sustain the life of the cell and, by extension, the life of the organism. As we reflect on the organelles, it is soothing to see the harmony they represent. They are small, intricate, and purposeful, carrying out their duties without pause, often without error. They remind us that life is not only about the grand and visible, but also about the quiet and unseen. Within each cell, hidden from view, organelles are at work, making possible every movement, every breath, every thought. In the next part, we will look more closely at the nucleus itself, the command center of the cell, and explore the way in which DNA, carefully stored within, carries the instructions for life. We will see how this genetic material is organized, how it is read, and how it ensures continuity from one cell to the next. In this way, we will come closer to the secret at the heart of the cell, the code of life itself. 9. The nucleus and DNA. At the center of nearly every eukaryotic cell lies a structure of profound importance, a quiet chamber that holds the instructions for life. This is the nucleus, often called the command center of the cell, for within its walls resides DNA, the molecule that carries the genetic code. Though it occupies only a small portion of the cell's volume, the nucleus governs nearly everything the cell does, from the proteins it produces to the way it grows, divides, and responds to its environment. It is both a library and a regulator, preserving the record of life while guiding its daily activity. The nucleus is enclosed by a double membrane known as the nuclear envelope. This barrier, dotted with thousands of small pores, separates the delicate contents of the nucleus from the surrounding cytoplasm. Yet it is not a closed box. The pores serve as gateways, allowing certain molecules to pass in and out. Proteins needed for the maintenance of DNA enter through these portals, while messenger RNAs, freshly transcribed from the genetic code, exit to guide protein synthesis elsewhere in the cell. This regulated traffic ensures that the nucleus remains protected, while still maintaining communication with the rest of the cell. Inside the nucleus, DNA is arranged with remarkable care. Rather than floating freely, it is wrapped around proteins called histones, forming structures known as chromatin. This packaging serves several purposes. It allows the long, fragile strands of DNA to fit within the confines of the nucleus, and it also helps regulate which parts of the genetic code are accessible at any given time. In regions where the DNA is tightly wound, the information is hidden and inactive. In regions where it is loosely arranged, the code is open and can be transcribed into RNA. This balance between open and closed chromatin is one of the ways a cell controls its functions, ensuring that the right genes are expressed at the right time. When a cell prepares to divide, the chromatin coils even more tightly, forming the familiar shapes of chromosomes. These condensed structures are easier to separate accurately, ensuring that each daughter cell receives a complete set of instructions. Humans, for example, carry 46 chromosomes in most of their cells. Each chromosome holds hundreds or even thousands of genes, sequences of DNA that serve as the blueprints for proteins. Together, these chromosomes contain the entirety of the human genome, the complete set of instructions for building and sustaining a human being. The DNA itself is a molecule of extraordinary design. Shaped as a double helix, it resembles a twisted ladder, with two strands winding around one another. The sides of the ladder are made of sugar and phosphate molecules, while the rungs are pairs of chemical bases. There are 4 types of bases: adenine, thymine, cytosine, and guanine, and they pair in specific ways: adenine with thymine, cytosine with guanine. The order of these bases along the strand forms the genetic code, a sequence of letters that spells out the instructions for life. Just as letters in a sentence can be arranged to create meaning, so too can the sequence of bases create the instructions for building proteins. The nucleus not only stores this information but also manages its use. When a cell needs to produce a particular protein, the relevant section of DNA is copied into a temporary molecule called messenger RNA. This process, known as transcription, is carried out by enzymes that move along the DNA strand, reading its code and producing a complementary RNA sequence. The messenger RNA then leaves the nucleus through a pore and enters the cytoplasm, where ribosomes use its instructions to build the protein. In this way, the nucleus directs the cell's activities by choosing which genes are expressed, when, and in what amounts. Within the nucleus is another structure, the nucleolus, which appears as a darker, denser region under a microscope. The nucleolus is not enclosed by its own membrane, but it serves an important purpose. It is the site where ribosomal RNA is synthesized and combined with proteins to form ribosomal subunits. These subunits later assemble into ribosomes in the cytoplasm, the machines that build proteins. The nucleolus, therefore, plays a central role in ensuring that the cell has the tools it needs to carry out protein synthesis. The importance of DNA within the nucleus extends beyond the life of a single cell. When cells divide, they must pass their DNA on to their daughter cells. This continuity ensures that the new cells carry the same instructions, maintaining the identity and function of tissues. It also allows inheritance across generations, as DNA from parents is passed to offspring. The nucleus, in this sense, is not only a command center for daily operations, but also a vessel of continuity, carrying the blueprint of life from past to future. It is calming to reflect on how orderly this process is. Within each cell, strands of DNA several meters long are folded into a nucleus, only a few micrometers wide. The instructions within those strands guide the building of proteins, the activity of enzymes, the growth of tissues, and the balance of entire organisms. Despite the staggering complexity of these instructions, the system unfolds smoothly, with transcription, translation, and regulation occurring in quiet cycles that sustain life moment by moment. There is also a beauty in the way the nucleus allows diversity. Not every cell in the body uses the same parts of the DNA. Muscle cells focus on genes that enable contraction. Neurons focus on genes that allow signaling. Skin cells focus on genes that provide barriers and protection. Though every cell carries the full genome, each one expresses only the genes it needs. This selective use of DNA allows cells to specialize, forming tissues and organs that together create the complexity of the human body. The nucleus, through its control of gene expression, makes this specialization possible. As you picture the nucleus, you might imagine a quiet library filled with scrolls, each scroll holding instructions for a different task. The doors of the library are carefully guarded, allowing messages to leave when needed, but protecting the scrolls from harm. Some scrolls are read often, their messages copied daily. Others remain closed, perhaps never to be read in a particular cell. The library itself never rests. It is constantly balancing preservation with activity, continuity with change. In the end, the nucleus is both a symbol and a reality of life's organization. It holds the code that defines each organism, ensures its faithful transmission, and regulates its expression with remarkable precision. Without the nucleus and the DNA within it, the intricate dance of life could not continue. It is the quiet center, the guiding voice, the thread of continuity that connects each cell to its past and its future. In the next part, we will move outward again, looking not only at the nucleus, but at the wider cycles of cell life. We will explore how cells grow, how they divide, and how they progress through stages of activity, ensuring renewal and continuity. In doing so, we will see how the nucleus and its DNA participate in one of the most fundamental rhythms of biology, the cell cycle. 10. The cell cycle. Every cell that lives must also renew itself, for no living system can endure without change. This process of renewal unfolds in a rhythm known as the cell cycle. It is the sequence of events by which a cell grows, prepares, duplicates its DNA, and eventually divides to form new cells. The cycle is not rushed, but carefully measured, guided by signals and checkpoints that ensure accuracy. In this way, the cell cycle is both a story of growth and of continuity, a quiet pattern that repeats itself endlessly in every living organism. The cycle begins with a phase of growth, when the cell focuses on expanding its size and carrying out its usual duties. This stage is sometimes called interphase, and though it appears uneventful under a microscope, it is a time of extraordinary activity. The cell is busy producing proteins, building organelles, and preparing the internal conditions it will need for division. During this phase, the DNA remains in its loosely packed form, chromatin, so that genes can be accessed easily and transcription can continue. It is a period of preparation, not of idleness, and it often occupies the majority of the cell's lifetime. As the cell progresses, it enters a crucial step. Every base pair, every gene, every chromosome must be copied with precision. This stage, called the synthesis phase, ensures that when the cell divides, each daughter cell receives a complete set of genetic instructions. The process is carried out by enzymes that move along the DNA, unwinding the strands and building new complementary strands. The accuracy of this replication is extraordinary, though errors occasionally occur. Cells possess proofreading mechanisms that detect and correct many of them, preserving the integrity of the genetic code. The copying of DNA is a moment of quiet continuity, ensuring that the story of life can be passed forward without interruption. After DNA is duplicated, the cell continues to prepare. It checks its own systems, ensuring that proteins are properly folded, that organelles are ready to be shared, and that the environment is suitable for division. These checks are not trivial. If damage is detected, the cycle can pause while repairs are made. If the damage is too great, the cell may trigger a process of self-destruction known as apoptosis, protecting the organism from the risk of passing on faulty information. In this way, the cell cycle contains safeguards, mechanisms of patience and caution that ensure fidelity across generations. Eventually, the cell enters mitosis, the phase of visible division. During this stage, the chromatin condenses into distinct chromosomes, each a carefully coiled copy of DNA. The nuclear envelope dissolves, allowing the chromosomes to move freely. Structures called spindle fibers form, attaching to the chromosomes and guiding their movement. One by one, the chromosomes align at the center of the cell, and then, with perfect coordination, they are pulled apart, each half moving to opposite sides. This separation ensures that both daughter cells will inherit identical sets of genetic material. The cell then begins to constrict, its membrane pinching inward until it divides fully into two new cells. Each of these cells, though smaller, is complete, carrying the full potential of life within it. The entire process is a cycle. For once division is complete, the new cells begin again with growth, activity, and preparation. Some cells move quickly through the cycle, dividing often. Skin cells, for example, renew themselves frequently, forming new layers to replace those that are shed. Blood cells are also replenished regularly, ensuring that the body maintains balance. Other cells, such as certain nerve cells, exit the cycle and remain in a resting state, carrying out their functions for years without division. The variation in cycle length reflects the diverse needs of the organism. It is soothing to consider how many times this cycle is unfolding at this very moment. In the human body alone, billions of cells divide every day, each one faithfully copying DNA, aligning chromosomes, and forming new life. The scale is almost unimaginable, and yet it happens with such precision that we rarely notice. Without the cell cycle, wounds would not heal, growth would not occur, and continuity of life would falter. With it, the body maintains itself, generation after generation, cell after cell. The cycle also reflects a larger truth about life, that change and continuity are intertwined. Each division creates something new, a daughter cell distinct in time and space from its parent. Yet each division also preserves the essential code, ensuring that the lineage of cells remains unbroken. This balance between renewal and preservation is one of the deepest rhythms of biology, a quiet pulse that sustains all living things. The cell cycle is not only a process of mechanics, but also a process of harmony. It is governed by signals within the cell and by messages from the surrounding tissue. Growth factors, proteins, and enzymes all communicate, deciding when the cycle should proceed and when it should pause. This coordination ensures that cells divide only when needed, maintaining the health of the organism. In this way, the cycle reflects both independence and cooperation, for each cell is guided by its own machinery, yet also listens to the larger needs of the body. As you reflect on the cell cycle, it may help to imagine a gentle circle, always turning, always repeating. Within this circle are moments of activity, of copying, of separation, and of renewal. There are also moments of rest, of waiting, of caution. Together, they form a rhythm that is steady and unending, like a heartbeat at the smallest scale. Each turn of the cycle carries forward the essence of life, ensuring that cells endure, that organisms grow, and that generations continue. In the next part, we will look at how cells communicate with one another, sending and receiving signals that allow tissues and organs to coordinate their activities. This communication, subtle yet vital, reveals how cells do not act alone but as members of a greater community, each listening and responding to the others in the quiet conversation of life. 11. Cell communication. Life at the level of the cell is not solitary, though each cell is capable of carrying out its own functions. Survival in a body or in a community of organisms depends on the ability to communicate. Cells must share information, respond to signals, and coordinate their actions so that tissues and organs can function as one. This communication is subtle, often invisible, but it is constant, a quiet conversation that never ceases. Without it, the balance of life would quickly unravel. Communication between cells takes many forms. Some signals travel only short distances, passing from one neighboring cell to the next. Others travel farther, carried through the bloodstream or the surrounding fluid to reach cells scattered across the body. Still others remain entirely within the cell itself, small messages that guide internal processes. Whatever the distance, the principle is the same. A signal is sent, it is received, and a response follows. In this way, cells listen, speak, and adapt in harmony with their environment. At the surface of the cell, the membrane plays a crucial role in this conversation. Scattered across it are proteins that act as receptors. Each receptor has a shape that matches a specific signal, much like a lock fits a particular key. When the signal arrives, perhaps in the form of a hormone, a neurotransmitter, or a small molecule, it binds to the receptor, and the receptor changes shape. This change sets off a cascade of events inside the cell, a chain reaction that alters behavior. The binding of a single signal on the surface can influence the activity of enzymes, the movement of ions, the expression of genes, or the release of other signals. Consider, for example, the communication between nerve cells. At the junction between two neurons, one cell releases molecules called neurotransmitters. These molecules drift across the tiny gap and bind to receptors on the neighboring cell. The binding opens ion channels, allowing charged particles to flow, creating an electrical impulse that continues the message. In this way, thoughts are formed, muscles are commanded, and senses are perceived. The conversation is rapid, yet it is built on the same principles of signaling and reception found in all cells. Other forms of communication are slower but no less important. Hormones, produced in specialized glands, travel through the bloodstream to distant cells. Insulin, for instance, is released when blood sugar levels rise. It travels to cells throughout the body, instructing them to take in glucose and store it. This coordination ensures that energy is distributed evenly, that no part of the body is overwhelmed, and that balance is maintained. Such long-distance communication shows how even the smallest cells are participants in the larger rhythm of the whole organism. Cells also speak directly to their neighbors through physical contact. In tissues, cells are often connected by junctions, small channels that allow molecules to pass from one cell to another. Through these channels, ions and signaling molecules move freely, synchronizing activity. In the heart, for instance, such junctions allow waves of ions to pass quickly from cell to cell, ensuring that the muscle contracts in unison. This cooperation creates the steady beating of the heart, a rhythm sustained by countless tiny conversations. Inside the cell, communication is equally vital. Signals from the environment must be interpreted and passed inward, where they influence the nucleus and the expression of genes. This process is known as signal transduction, and it often involves multiple steps. A signal at the membrane activates a receptor. The receptor activates an enzyme. The enzyme produces small messenger molecules, and these messengers spread the signal throughout the cytoplasm. In this way, a single event at the surface can reach deep into the cell, guiding its choices. The cell's ability to amplify and integrate signals allows it to respond with precision, neither overreacting nor ignoring important information. Communication is also essential for the coordination of growth and division. Cells must know when to divide and when to remain still. Growth factors, small proteins secreted by other cells, bind to receptors and encourage entry into the cell cycle. If these signals are absent, the cell rests. If they are present, the cycle continues. In this way, tissues grow when needed, as during development or wound healing, but remain stable at other times. The balance of these signals protects the body from uncontrolled growth, reminding us again that communication is as much about restraint as it is about action. The immune system provides another striking example of cellular conversation. When a pathogen enters the body, certain cells detect its presence and release chemical signals. These signals summon other immune cells, instructing them to arrive at the site of infection. Once there, additional signals are exchanged, coordinating the attack. Some cells engulf the invader, others release antibodies, and others remember the encounter for future protection. At every step, communication guides the process, ensuring that the defense is effective and targeted. Without this exchange of messages, the immune system would be blind and disorganized. There is also a quieter form of communication, one that occurs when a cell speaks to itself. This is known as autocrine signaling, where the cell releases a molecule that binds to its own receptors. By doing so, it reinforces its decisions, strengthening its own signals. This type of self-talk may seem redundant, but it provides stability, ensuring that the cell's internal state matches its external behavior. Even at the smallest scale, life values consistency and reassurance. As you reflect on these patterns of communication, it is soothing to imagine the body as a community of cells engaged in a constant, gentle conversation. Each cell listens to its neighbors, responds to distant calls, and sends messages of its own. The conversation is orderly, respectful, and purposeful, with every signal contributing to the balance of the whole. The cells do not compete for attention, but instead harmonize their actions, maintaining the quiet symphony of life. This perspective reminds us that even at the smallest scale, connection is essential. No cell exists in isolation, and no function can be carried out alone. From the fastest impulse in a nerve to the slow release of a hormone, communication is the thread that binds the body together. It is subtle, invisible, and unceasing, the language through which life sustains itself. In the next part, we will turn our attention to energy once more, focusing on how cells obtain, store, and use it. By looking more closely at processes such as cellular respiration and photosynthesis, we will see how the energy of the environment is transformed into the fuel of life, flowing through cells in quiet cycles that never end. 12. Cellular energy. At the heart of every living process lies energy. Without energy, the quiet work of the cell could not unfold. No proteins would be built, no signals would pass, no division would occur. The cell is alive only because it is able to capture, transform, and use energy in an endless cycle. Unlike the sudden surges of force we sometimes imagine in the physical world, cellular energy flows steadily and gently, in countless small reactions that together sustain life. To reflect on cellular energy is to reflect on the quiet fire that burns within all living things. The molecule most often associated with this energy is adenosine triphosphate, or ATP. It is sometimes called the energy currency of the cell, for it carries small packets of power that can be spent on a wide variety of tasks. ATP consists of a chain of 3 phosphate groups attached to a core molecule. The bonds that hold the last phosphate in place are rich in potential energy, and when one of these bonds is broken, energy is released. The molecule then becomes adenosine diphosphate, or ADP, which can be recycled by adding another phosphate once more. In this way, ATP is continually spent and renewed, a gentle cycle of use and replenishment. To understand how cells maintain their supply of ATP, it is helpful to look at the process of cellular respiration. This is the set of reactions by which nutrients, particularly glucose, are broken down to release energy. The process begins in the cytoplasm, where glucose is split into smaller molecules in a series of reactions known as glycolysis. This step produces a modest amount of ATP, as well as molecules that carry electrons to later stages. The products then enter the mitochondria, where the majority of energy extraction takes place. Inside the mitochondria, the pathway continues through the citric acid cycle, sometimes called the Krebs cycle. Here, the smaller molecules, derived from glucose, are further broken down, releasing carbon dioxide as a byproduct. As bonds are broken, high-energy electrons are transferred to carrier molecules. These carriers then deliver the electrons to the electron transport chain, a series of protein complexes embedded in the inner membrane of the mitochondrion. It is along this chain that the greatest transformation occurs. The electrons move step by step through the chain, and at each step, their energy is used to pump protons across the membrane, creating a gradient. This gradient represents stored potential, like water held behind a dam. When the protons flow back across the membrane, they pass through an enzyme called ATP synthase, which harnesses the energy to form new ATP molecules. The process is quiet, precise, and continuous, yielding far more ATP than earlier stages. Oxygen is the final recipient of the electrons, combining with protons to form water. In this way, respiration depends on oxygen, and oxygen becomes essential for life as we know it. Not all cells, however, rely solely on oxygen. Some are capable of fermentation, a process that extracts energy from glucose in the absence of oxygen. Fermentation yields far less ATP than respiration, but it provides a survival strategy in environments where oxygen is scarce. In human muscles, for example, fermentation occurs when exertion outpaces oxygen delivery, producing lactic acid as a temporary product. Though less efficient, this pathway demonstrates the adaptability of cells, their ability to find energy in varied conditions. In plants, energy enters the cycle in a different way through the process of photosynthesis. Within the green structures of chloroplasts, light from the sun is captured and transformed into chemical energy. Pigments, such as chlorophyll, absorb the energy of photons, exciting electrons that drive a chain of reactions. Water molecules are split in the process, releasing oxygen into the air. The energy is stored first in molecules like ATP and NADPH, then used to build glucose from carbon dioxide. This glucose becomes the fuel not only for the plant itself, but also for the countless other organisms that depend on plants for nourishment. In this sense, photosynthesis and respiration are two halves of a larger cycle. The oxygen released by plants sustains respiration in animals, while the carbon dioxide exhaled by animals sustains photosynthesis in plants. Energy in the cell is not only about capturing and storing, but also about using it wisely. ATP powers muscle contraction, allowing fibers to slide past one another in smooth motion. It fuels active transport, the pumping of ions and molecules across membranes against their natural gradients. It drives chemical reactions that would not otherwise occur, making possible the synthesis of complex molecules from simple ones. Each act of energy use is small, but taken together, They create the activity of life. The flow of energy is constant. At any moment, a molecule of ATP may be consumed, releasing its phosphate and powering a reaction, only to be rebuilt moments later in the mitochondrion. In the human body, the total supply of ATP is small, yet it is turned over many times each day, replenished as quickly as it is spent. This ceaseless cycle is invisible, but it is as steady and reliable as breathing. It is the hidden current beneath every movement, every thought, every quiet process that unfolds within. There is something soothing in recognizing the symmetry of energy in life. The energy that fuels a human being comes from the food they eat, food that once grew through sunlight and soil, food that was itself built from the energy of photosynthesis. That same energy, captured long ago by plants, flows into muscles, nerves, and organs, and is eventually released as heat. The cycle then begins again. As new plants grow, new sunlight is captured, and new food is formed. Energy is never created or destroyed, only transformed, and the cell is the site of these transformations, endlessly repeating. If we pause and imagine this process, it becomes a picture of quiet balance. In the cytoplasm, glucose is split. In the mitochondria, electrons flow along the chain. In the chloroplast, light is absorbed and turned into sugar. Each step is small, and yet together they sustain the continuity of life. There is no pause in this work, no moment when energy ceases to flow. It is the gentle hum of existence, the constant spark that underlies all living things. In the next part, we will explore how cells organize themselves into tissues, working not as isolated units but as members of a greater whole. Through cooperation and specialization, cells create the structures of the body, and by studying tissues, we begin to see how cellular life gives rise to the complexity of organisms. 13. Tissues and organization. When we look at a single cell under a microscope, it can feel like a complete and self-sufficient world. It has its own boundary, its own energy, its own instructions, and its own machinery for survival. Yet, within the human body, cells rarely live in isolation. Instead, they come together in groups, cooperating and specializing, forming patterns that create the structure of tissues. These tissues, in turn, combine to form organs, and organs join into systems until the complexity of the human body emerges. The transition from single cells to tissues is one of the most beautiful demonstrations of how unity and cooperation create strength in life. A tissue can be thought of as a community of similar cells that share both structure and function. There are 4 main categories in the human body: epithelial, connective, muscle, and nervous tissue. Each has its own unique qualities, and each contributes in a particular way to the functioning of the body. Though they differ in appearance and behavior, they share one unifying principle. No tissue works entirely alone, and no cell within a tissue works without regard for its neighbors. Together, they form the fabric of life. Epithelial tissue forms the coverings and linings of the body. It can be found on the surface of the skin, lining the digestive tract, covering organs, and forming the delicate boundaries of glands. Its cells are tightly packed, forming continuous sheets that act as barriers. Some epithelia are specialized for absorption, drawing nutrients into the body. Others are devoted to secretion, releasing enzymes, hormones, or mucus. Still others provide protection, shielding the body from the outside world. Despite their different roles, all epithelia share a simple quality. They create interfaces between the inner and outer worlds, mediating the exchange of substances and signals with careful precision. Connective tissue, as its name suggests, provides support and connection. It is one of the most diverse categories, encompassing bone, cartilage, blood, fat, and the fibrous tissues that hold organs together. In connective tissue, cells are often scattered within an extracellular matrix, a substance that they themselves produce. This matrix may be hard, as in bone, flexible, as in cartilage, or fluid, as in blood. Through these variations, connective tissue gives the body strength, elasticity, and resilience. It binds structures, cushions organs, stores energy, and even transports oxygen and nutrients. Though often working in the background, connective tissue is essential, the framework upon which the body rests. Muscle tissue is specialized for movement. It is composed of cells capable of contracting, pulling against one another to create force. There are 3 types of muscle tissue: skeletal, cardiac, and smooth. Skeletal muscle attaches to bones and produces voluntary movements, allowing us to walk, speak, and reach. Cardiac muscle is found only in the heart, and its rhythmic contractions pump blood through the body without pause. Smooth muscle lines the walls of internal organs, gently moving food through the digestive tract or regulating the diameter of blood vessels. The work of muscle tissue is constant, whether in the deliberate movement of limbs or in the quiet contractions that sustain life without conscious thought. Nervous tissue forms the body's communication system. It is composed of neurons, specialized cells that can generate and transmit electrical impulses, and of supporting cells that nourish and protect them. Neurons carry information across long distances, connecting sensory input to thought and thought to action. Within nervous tissue, information flows in patterns of extraordinary complexity, guiding everything from reflexes to reasoning. Though neurons may seem far removed from the simple image of a cell, they are built from the same foundations, and they too depend on the same quiet rhythms of energy and signaling. Nervous tissue is the conductor of the body's orchestra, allowing countless cells and tissues to act in harmony. The cooperation of these tissues becomes clear when we consider the structure of organs. Take, for example, the stomach. Its surface is lined with epithelial cells that secrete mucus, enzymes, and acid. Beneath this layer, smooth muscle cells contract rhythmically, churning food. Connective tissue supports the structure, providing elasticity and strength. Nervous tissue coordinates the contractions and secretions, ensuring that digestion proceeds in an orderly way. Each tissue contributes, and only together does the stomach function. The same is true for every organ, from the heart to the lungs, from the skin to the brain. They are mosaics of tissues, assembled from cells, working as one. It is easy to think of the body in terms of its grand systems: the circulatory system, the digestive system, the nervous system. But at its foundation, all of these are simply layers of organization built upon the cooperation of cells. Cells form tissues, tissues form organs, organs form systems, and together, these systems form the organism. This nested structure allows for complexity, but it also ensures stability. Each level builds upon the one before it, and each level depends on the quiet functioning of the smallest unit, the cell. The organization of cells into tissues also allows for specialization. A single cell cannot do everything. It cannot simultaneously be strong, contractile, protective, and communicative. But groups of cells can divide the labor. Some focus on contraction, others on support, others on signaling, others on defense. This division of labor mirrors what we see in human communities, where individuals with different skills contribute to a shared purpose. Within tissues, the same principle applies, creating a body that is more than the sum of its parts. There is a deep comfort in reflecting on this organization. The tissues of the body do not compete with one another, but instead complement one another. The skin protects, the muscles move, the bones support, the nerves guide, the blood nourishes. Each has its place, each plays its role, and together they sustain life. The complexity may seem vast, but at its core, it is an expression of cooperation and harmony. As you picture this organization, you may imagine a quiet weaving of threads into fabric. Each thread on its own is fragile, but when combined with others, it forms something strong and resilient. Tissues are such weavings, threads of cells intertwined into patterns that support the body. The organs are garments made of these fabrics, each tailored for a particular function, and the systems of the body are like the whole attire, complete and coordinated, allowing the organism to meet the world with grace and strength. In the next part, we will explore how cells and tissues maintain balance, a concept known as homeostasis. We will see how the body regulates temperature, pH, and energy levels, ensuring that every cell can continue its work. This constant balance, though often unnoticed, is one of the most profound features of life, a steady calmness that sustains existence from moment to moment. 14. Homeostasis. Life, in all its complexity, depends on a remarkable constancy. Though the environment outside may change, temperatures rise and fall, food is abundant or scarce, light fades into darkness, the interior of the body remains stable. This steady state, maintained by countless small adjustments, is known as homeostasis. It is the balance that allows cells to continue their work without interruption, the calm background against which the drama of living unfolds. Without homeostasis, the intricate machinery of the cell could not function, for even the smallest deviation from balance can disturb the delicate chemistry upon which life depends. At its essence, homeostasis is about regulation. The body continuously monitors its internal conditions, comparing them against set points, and when variation occurs, it responds with corrections. Temperature is regulated, pH is buffered, sugar levels are adjusted, oxygen is delivered, and wastes are removed. Yet together they create stability. Cells thrive not in chaos, but in environments where conditions remain within narrow ranges. Homeostasis is the reason they can carry on quietly, building, signaling, dividing, and renewing. Temperature provides one of the clearest examples. Human cells function best near 37 degrees Celsius, and though the world outside may swing from freezing cold to intense heat, the body maintains this internal warmth with remarkable precision. When the temperature rises, sweat glands release moisture, and as it evaporates, heat is carried away. Blood vessels in the skin dilate, allowing more blood to flow near the surface where heat can escape. When the temperature falls, muscles contract in small shivers, generating warmth, and blood vessels constrict to preserve heat within. These responses are automatic, carried out by networks of signals and effectors, ensuring that the cellular environment remains constant no matter the weather. The balance of pH is equally important. Most cellular reactions occur best in conditions that are slightly alkaline, around a pH of 7.4. If the blood becomes too acidic or too basic, enzymes falter and processes slow or fail. To prevent this, the body uses buffers, chemical systems that absorb excess hydrogen or hydroxide ions and keep pH within range. The lungs also contribute by adjusting the amount of carbon dioxide in the blood, and the kidneys excrete acids or bases as needed. Together, these systems maintain a delicate equilibrium, protecting cells from shifts that could disrupt their work. Glucose regulation offers another view of homeostasis, one deeply tied to cellular energy. Cells depend on glucose as a fuel, but the levels in the bloodstream must be kept steady. After a meal, glucose levels rise, and in response, the pancreas releases insulin. This hormone signals cells to absorb glucose, storing it as glycogen or fat for later use. When glucose levels fall, another hormone, glucagon, prompts the release of stored energy back into the blood. This balance ensures that cells always have the fuel they require, avoiding both excess and scarcity. It is a quiet cycle of abundance and conservation, always adjusting, always returning to the middle ground. Oxygen and carbon dioxide provide yet another example. Cells need a constant supply of oxygen for respiration, and they must also release carbon dioxide, a byproduct of metabolism. The body maintains this balance through breathing and circulation. Sensors in the blood detect oxygen and carbon dioxide levels, sending signals to the brainstem, which adjusts breathing rate accordingly. At the same time, the heart pumps blood, delivering oxygen-rich cells to every corner of the body and carrying away waste gases. The rhythm of breath and the beat of the heart are both instruments of homeostasis, keeping the internal environment steady and favorable for life. Homeostasis is not carried out by one system alone, but by cooperation across all levels of the body. The nervous system detects changes quickly and coordinates rapid responses. The endocrine system, through hormones, adjusts processes more gradually, but with longer-lasting effects. The kidneys, lungs, skin, and digestive tract all play roles in excretion, absorption, and regulation. Together, these systems form a network of guardians, each tuned to preserve balance, each acting in concert with the others. What makes homeostasis especially remarkable is its subtlety. It is not an absolute stillness, but a dynamic equilibrium. Conditions do not remain perfectly fixed, but fluctuate gently within ranges. Blood sugar rises after eating, then falls again. Temperature drifts slightly with activity, then steadies. Breathing quickens during exertion, then slows in rest. These oscillations are not signs of failure, but of responsiveness, evidence that the body adjusts itself continually. In this way, homeostasis is less like a rigid wall and more like a steadying hand, keeping balance through constant small movements. Cells themselves also contribute to their own homeostasis. Membranes regulate what enters and exits. Organelles recycle and detoxify. Enzymes adjust activity with temperature and pH. Each cell is a microcosm of regulation, maintaining its internal environment, while relying on the larger body to keep the external conditions favorable. The harmony between cellular and systemic homeostasis ensures survival, from the level of molecules to the level of the whole organism. There is something deeply calming in recognizing the quiet persistence of these processes. As we go about our days, unaware, our bodies are constantly adjusting, compensating, and rebalancing. The heart changes its pace, the lungs alter their rhythm, the kidneys fine-tune their excretions, the skin sweats or conserves heat. All of this unfolds without effort on our part, a silent orchestration that allows life to continue smoothly. Homeostasis is the background music of existence, steady and unbroken. It is also a reminder of resilience. Though disturbances occur, illness, stress, environmental extremes, the body works tirelessly to restore balance. Sometimes the challenge is great, and balance is only partially maintained. But the effort persists, for survival depends on it. This resilience is built into the very fabric of life, a testament to the strength of cells and systems that have evolved to endure change while preserving continuity. In the end, homeostasis is more than a biological process. It is a reflection of life's preference for balance, for harmony, for the quiet middle ground. It is the reason cells can thrive in a world of flux, the reason organisms can grow, adapt, and continue. It is the gentle constancy that underlies every breath, every heartbeat, every moment of being. In the next part, we will look at how cells age and what happens as the balance of homeostasis gradually shifts with time. By exploring cellular aging, we will see how the quiet processes of life unfold not only in cycles of renewal, but also in gentle decline, and how even in aging, there is continuity and calm. 15. Cellular aging. Every living cell carries within it the quiet rhythms of time. From the moment it is born, a cell is engaged in a cycle of activity, growth, division, and repair. Yet, like all things in nature, cells do not endure forever in their prime. Over the course of days, months, or years, they begin to change, gradually losing some of their vitality. This process is known as cellular aging. It is not a sudden shift, but a gentle progression, shaped by countless small events and choices made by the cell. To reflect on cellular aging is to reflect on the way life unfolds in cycles, beginning, flourishing, and eventually slowing, always with calm continuity. One of the most significant factors in cellular aging is the gradual shortening of structures called telomeres. Telomeres are stretches of repetitive DNA that sit at the ends of chromosomes, protecting the important genetic information within. Each time a cell divides, the DNA must be copied. But the very ends of the chromosomes cannot be replicated perfectly. As a result, telomeres grow shorter with each division. At first, this shortening has little effect, for the telomeres serve as protective caps. But eventually, when the telomeres become too short, the cell can no longer divide safely. At this point, the cell may enter a state of rest, known as senescence, or it may be guided toward a natural end. In this way, telomeres act like biological clocks, measuring the passage of time within each cell. Beyond telomeres, the wear and tear of daily life also contributes to aging. Every moment, cells are exposed to byproducts of their own metabolism. Oxygen, so essential for energy, can sometimes form reactive molecules that damage DNA, proteins, and membranes. These reactive oxygen species are usually neutralized by antioxidants, but not always completely. Over the years, the small accumulations of damage add up, gradually reducing the efficiency of cellular machinery. Enzymes may fold less reliably, membranes may lose flexibility, and DNA may carry scars of past insults. The cell still functions, but with a little less vigor than before. Mitochondria, the powerhouses of the cell, also play a role in aging. These organelles generate the energy that sustains life, but in doing so, they produce reactive molecules that can damage their own DNA. Unlike nuclear DNA, which is carefully protected, mitochondrial DNA is more exposed and more vulnerable. Over time, errors accumulate, and mitochondria may become less efficient at producing energy. As this efficiency declines, the cell has less fuel for its activities, and its overall vitality diminishes. The quiet hum of energy that once powered the cell begins to soften, a gentle slowing of life's rhythm. The cell's systems of repair also influence aging. Throughout its life, a cell encounters mistakes and damage, and it has many tools for correcting them. Enzymes patrol DNA for errors. Misfolded proteins are refolded or destroyed, and organelles are recycled through autophagy. But these systems, like all others, are not perfect. Over time, their effectiveness wanes, errors slip through more often, and damage accumulates. The balance tips gradually, from repair to decline, until the cell carries more imperfections than it can manage. This gentle accumulation is one of the hallmarks of aging. Senescence is a state that many aging cells eventually enter. In this condition, the cell no longer divides, but it does not die immediately. Instead, it remains metabolically active, releasing signals into its environment. Some of these signals are beneficial, helping with wound healing or limiting uncontrolled growth. But if too many senescent cells build up, they can contribute to inflammation and tissue decline. Senescence, then, is both protective and problematic. A natural pause in the cell's life cycle that helps the organism in one way, but hinders it in another. Not all cells age in the same way. Some, like red blood cells, live only for a short time before being replaced. Others, like certain nerve cells, may endure for decades, carrying memories and functions across a lifetime. Stem cells, which have the ability to renew and produce many other cell types, also age, though they do so more slowly. Their decline can influence the body's ability to regenerate and repair. The diversity of aging reflects the diversity of life, with each cell type following its own path, yet all guided by the same underlying principles of time and change. It is important to see that cellular aging is not simply loss, but also adaptation. Cells that can no longer divide may still contribute to the health of tissues in subtle ways. The gradual slowing of processes reflects not failure, but balance, for unchecked growth can be just as dangerous as decline. In this sense, aging is one of life's strategies, a way of ensuring stability over time. It is not a sudden collapse, but a gentle modulation, a rebalancing that shapes the course of life. There is a certain peace in acknowledging the inevitability of cellular aging. Just as leaves on a tree grow, flourish, and eventually fall, so too do cells move through their cycles of vigor and rest. The body as a whole remains alive and vital because of the constant renewal of cells. But the aging of individual cells is part of the larger story. It allows space for replacement, for adaptation, and for the unfolding of life in its natural rhythm. As we reflect on cellular aging, it becomes clear that time is woven into the fabric of biology. Every division, every repair, every breath of oxygen leaves its trace. The quiet shortening of telomeres, the subtle accumulation of wear, the soft fading of energy, all are reminders that life moves forward in gentle steps. And yet, even as individual cells age, the organism endures, carried forward by new cells, new growth, and new cycles of renewal. In the next part, we will look at how cells sometimes end their journeys altogether, not through aging alone, but through programmed cell death, a process called apoptosis. By exploring this natural conclusion, we will see how even the end of a cell's life is guided by order and balance, contributing to the health of the whole. 16. Apoptosis and cellular renewal. In the quiet life of the cell, there comes a time when existence must draw to a close. Yet in biology, even endings are carried out with care and with order. Cells do not merely cease to function. Many follow a path of programmed departure known as apoptosis. This process, often called programmed cell death, is not violent or chaotic. It is a carefully orchestrated sequence of events, ensuring that the end of one cell contributes to the health and renewal of the whole organism. In apoptosis, we see a profound truth of life, that endings are also beginnings, and that through the death of some cells, the body maintains balance and continuity. Apoptosis differs greatly from accidental death, which might occur from injury or sudden stress. In those situations, a cell may burst and spill its contents, causing inflammation and distress in surrounding tissue. Apoptosis, by contrast, is calm and orderly. The cell itself initiates the process, shrinking gently, breaking down its components in a controlled manner, and packaging them into small fragments. These fragments are then cleared away by neighboring cells or by specialized immune cells, leaving no trace of disruption. It is as though the cell tidies its space before quietly departing. The signals that trigger apoptosis come from both within and outside the cell. Inside, damage to DNA, malfunctioning organelles, or excessive stress can activate pathways that lead to self-destruction. Outside, signals from neighboring cells or from the immune system can encourage apoptosis when a cell is no longer needed or when it poses a risk. These pathways converge on a set of proteins that act like switches, turning on enzymes called caspases. Once activated, caspases carefully dismantle the cell, breaking down proteins, cutting DNA, and disassembling structures with precision. The process is irreversible, but it is also controlled, ensuring that the cell departs in harmony rather than disorder. Apoptosis plays a vital role in development. During embryonic growth, for example, apoptosis shapes the form of the body. In the early stages, a developing hand resembles a paddle. Through apoptosis, the cells between the fingers are removed, sculpting the delicate spaces that define the hand. Similar processes occur throughout the body, carving structures, refining features, and ensuring proper function. Without apoptosis, the body would not achieve its form, and development would be incomplete. In adulthood, apoptosis continues to serve, maintaining balance by removing cells that are damaged, unneeded, or potentially harmful. Cells that have suffered irreparable DNA damage may choose apoptosis to prevent passing errors to their descendants. Cells infected by viruses may self-destruct to protect the organism. Even cells that have simply fulfilled their purpose may be guided gently to their end, making space for renewal. This constant turnover keeps tissues healthy, preventing the buildup of malfunctioning cells and ensuring that fresh cells can take their place. Apoptosis also works hand in hand with renewal. As old cells are removed, stem cells and other dividing cells step forward to create replacements. The skin, for example, is continually renewed. Cells at the surface flake away, guided into their end, while new cells from beneath rise to replace them. The lining of the digestive tract, too, is constantly refreshed, with old cells removed and new ones produced in a steady cycle. This balance between apoptosis and renewal creates a dynamic equilibrium, allowing the body to remain whole while individual cells come and go. The elegance of apoptosis is found not only in its biological purpose but also in its quiet symbolism. It is a reminder that endings need not be destructive, that closure can be a gentle process that makes way for growth. Just as the falling of leaves in autumn makes space for the new buds of spring, the departure of cells through apoptosis allows the body to remain healthy, resilient. It is a cycle of loss and renewal, woven into the very fabric of life. On a molecular level, the choreography of apoptosis is intricate. Membranes change their shape, signaling to immune cells that clearance is needed. DNA fragments into precise pieces, preventing confusion or error. Organelles are packaged neatly, their contents contained until they can be recycled. Neighboring cells, or phagocytes, recognize the signals and embrace the fragments, removing them without fuss. The result is a seamless transition, one cell leaving, another arriving, with the larger system undisturbed. Apoptosis is also a safeguard against danger. When cells refuse to follow the path of programmed death, they can accumulate mutations and grow uncontrollably. In such cases, the harmony of the body is threatened. By maintaining apoptosis, the organism preserves balance, ensuring that no single cell grows beyond its place. In this way, apoptosis is both a conclusion and a defense, a quiet gatekeeper protecting the whole. It is soothing to reflect on how much of life depends on such silent, unseen processes. At any moment, countless cells within the body are undergoing apoptosis, completing their journeys with dignity and order. At the same time, countless others are being born through division, carrying forward the work of life. The balance between these two movements, death and renewal, sustains us, though we rarely feel it or notice it. Beneath the surface of awareness, the cycle continues, moment by moment, cell by cell. As you imagine this cycle, you may picture a quiet forest. Old leaves fall gently to the ground, returning their nutrients to the soil. At the same time, new shoots emerge, reaching upward toward the light. The forest as a whole remains steady and alive, though its individual parts are constantly changing. In the same way, apoptosis and renewal create a landscape of balance within the body, where life persists through the graceful turnover of its smallest parts. In the next part, we will widen our view, looking at how cells interact with their environment and adapt to challenges. By exploring cellular responses to stress, we will see how cells not only endure but adjust, changing their behavior in subtle ways to protect themselves and to preserve the larger whole. 17. Cellular stress responses. Even in the most stable environments, cells encounter challenges. Nutrients may grow scarce, toxins may appear, temperatures may rise or fall, and oxygen may fluctuate. These shifts, though often subtle, place stress on the cell. Yet rather than collapsing in the face of difficulty, cells respond. They sense the change, adjust their processes, and adapt in ways that allow survival. These adjustments are known as stress responses, and they are among the most remarkable demonstrations of cellular resilience. In the quiet and hidden world within us, cells are constantly listening and adapting, maintaining balance even when conditions grow uncertain. One of the most common challenges cells face is oxidative stress. As cells produce energy, reactive oxygen species are sometimes formed. These molecules can damage DNA, proteins, and membranes, threatening stability. To counter this, cells deploy antioxidants, molecules that neutralize the reactive species and restore calm. Enzymes like superoxide dismutase and catalase play vital roles, converting harmful molecules into harmless water and oxygen. When antioxidant defenses are strong, the cell weathers oxidative stress with little harm. When they are overwhelmed, damage accumulates, reminding us that balance between stress and defense is always delicate. Heat stress offers another example. When temperatures rise beyond the usual range, proteins within the cell may unfold or misfold, losing their proper shapes. To manage this, cells produce heat shock proteins, specialized helpers that stabilize and refold proteins, preventing aggregation. These proteins act like careful attendants, guiding damaged molecules back into function or escorting them to recycling pathways. Through this mechanism, cells endure fevers, environmental heat, and other stresses that might otherwise threaten their integrity. The production of heat shock proteins shows how cells anticipate and manage danger with quiet efficiency. Nutrient deprivation also provokes adaptive responses. When glucose is scarce or amino acids limited, cells activate pathways that conserve energy and recycle internal components. One such pathway is autophagy, a process in which the cell encloses damaged organelles and proteins within membranes, then delivers them to lysosomes for breakdown. The resulting components are reused, providing fuel and building blocks. Autophagy is not only a survival strategy, but also a means of renewal, clearing away what is damaged and making space for what is fresh. In times of scarcity, autophagy allows the cell to endure, living more frugally while maintaining essential functions. Oxygen deprivation, known as hypoxia, presents another challenge. Cells rely on oxygen for respiration, and when it is limited, they shift their metabolism. Proteins called hypoxia-inducible factors accumulate, guiding the cell to reduce oxygen demand and increase the production of proteins that promote survival. In some cases, new blood vessels are encouraged to grow, delivering oxygen to tissues in need. This response shows the remarkable adaptability of cells, their ability to sense even subtle changes in oxygen and adjust their behavior accordingly. Stress responses also protect the integrity of genetic information. When DNA is damaged, cells activate repair pathways. Proteins scan the DNA, locate errors, and either fix them or signal for the cell cycle to pause until repairs are complete. If the damage cannot be repaired, apoptosis may be triggered, protecting the organism by removing a compromised cell. These responses ensure that the genetic blueprint is preserved as faithfully as possible, maintaining continuity across generations of cells. There are also unfolded protein responses, which occur when the endoplasmic reticulum becomes burdened with misfolded proteins. In such cases, sensors detect the accumulation and slow down protein synthesis, giving the cell time to recover. Chaperone proteins are increased, assisting in folding, and faulty proteins are sent for degradation. This stress response highlights the importance of quality control within the cell. Proteins must fold correctly to function, and the unfolded protein response ensures that errors are managed without overwhelming the system. The immune system also ties into cellular stress responses. Cells under strain often release signals to alert their neighbors or immune cells. These signals may call for assistance, trigger inflammation, or mark the stressed cell for removal. In this way, stress responses are not isolated, but part of a broader communication, ensuring that the whole organism can adapt together. What begins as a challenge in one cell can prompt coordinated adjustments across tissues, maintaining harmony. It is striking to realize how often stress responses succeed without our awareness. At any given moment, countless cells in the body are quietly adjusting to fluctuations in their environment, folding proteins, repairing DNA, neutralizing reactive molecules, or recycling their components. We may never feel these efforts, yet they sustain us, preserving function and balance in the face of change. The resilience of cells is one of the reasons organisms survive in a world that is never truly constant. There is a softness to the idea of stress responses, for they show not only vulnerability but also adaptability. Cells are not rigid structures that collapse under strain, but flexible, responsive systems. They bend without breaking, adjust without losing their essence, and endure without panic. In their quiet adjustments, they reflect a truth about life itself. That strength often lies in flexibility, in the ability to change gently without losing balance. As we imagine these processes, we might picture a calm lake with ripples forming on its surface. A gust of wind or a falling leaf disturbs the water, but the ripples soon fade, and the lake returns to calm. In the same way, stress disturbs the cell, but responses rise, balance is restored, and stillness returns. The underlying stability is not lost, only momentarily shifted, then gently regained. In the next part, we will move from stress responses to the idea of cellular specialization in greater depth, exploring how cells not only adapt but also differentiate, taking on unique identities that allow them to perform the diverse functions of the human body. Through differentiation, we will see how one set of instructions can give rise to endless variety, a quiet unfolding of diversity from a shared origin. 18. Cellular differentiation. All cells in the human body share a common origin, for they arise from the fertilized egg, a single cell that contains the full set of instructions for building an organism. From this one beginning, billions of cells are produced, each carrying the same genetic code. Yet, the body is not composed of identical units. Instead, cells acquire distinct shapes, structures, and functions, forming the many tissues and organs that allow life to flourish. This transformation, the process by which a cell becomes specialized for a particular role, is known as differentiation. It is one of the most extraordinary phenomena in biology, for it reveals how a single script can be read in countless ways, creating variety from unity. At the heart of differentiation lies the principle of selective expression. Every cell contains the entire genetic library, but no cell uses all of it at once. Instead, certain genes are activated while others remain silent. This selective turning on and off of genes determines the cell's character. A muscle cell expresses genes for contractile proteins, giving it the ability to move. A neuron expresses genes for channels and receptors, allowing it to transmit signals. A red blood cell turns on genes for hemoglobin, enabling it to carry oxygen. The differences in function arise not from differences in DNA, but from differences in the way the DNA is read. This selective expression is guided by signals both internal and external. Within the developing embryo, cells are exposed to gradients of chemical cues, subtle differences in concentration that provide positional information. Depending on where they are and which signals they receive, cells make choices about their futures. Some commit to becoming muscle, others to becoming skin, others to forming nerves or blood. These choices are stabilized by changes in the way DNA is packaged and accessed. Proteins wrap DNA into chromatin, and chemical marks determine which regions are open for transcription and which remain closed. In this way, the cell locks in its identity, preserving its specialization across divisions. Stem cells illustrate the potential of differentiation most clearly. These are cells that retain the ability to become many different cell types. In the earliest stages of development, embryonic stem cells can give rise to nearly any tissue, a property called pluripotency. As development continues, stem cells become more restricted, producing only certain lineages. In the adult body, stem cells remain in specific niches, quietly renewing blood, skin, or gut lining as needed. Their ability to divide and to produce specialized descendants ensures that tissues can repair themselves, maintaining health across a lifetime. The variety produced through differentiation is immense. Consider the differences between a neuron and a white blood cell. The neuron may stretch out long, delicate extensions called axons and dendrites, designed for transmitting signals across distance. The white blood cell, by contrast, may roam freely through the blood and tissues, changing shape as it engulfs invaders. Both descend from the same original code, yet their forms and behaviors are utterly distinct. This diversity allows the human body to perform functions far beyond the capacity of any single cell type. Differentiation is not a rigid destiny, but a balanced pathway. While many cells commit fully to their specialized roles, others retain some flexibility. Certain stem cells can divide asymmetrically, producing one specialized daughter and one that remains a stem cell. In this way, even some mature cells, under special conditions, can be coaxed back toward a more flexible state. This plasticity has become a subject of deep study, offering insight into regeneration and healing. In tissues, the cooperation of differentiated cells creates harmony. In the skin, for example, basal stem cells divide to produce keratinocytes, which gradually mature and migrate upward, forming protective layers. In the blood, stem cells in the marrow give rise to red blood cells, white blood cells, and platelets, each serving a different but essential purpose. In the nervous system, billions of neurons differentiate into countless subtypes, forming intricate networks of communication. Each system depends on the variety produced by differentiation, and each system thrives because these varieties work together. Differentiation also reveals the deep economy of life. Rather than writing separate genetic codes for each type of cell, evolution has written one shared script and allows it to be interpreted in many ways. This strategy not only conserves information but also ensures coherence. Because all cells share the same underlying code, they can communicate and coordinate more effectively. The diversity of form and function emerges not from separation, but from variation upon a common theme. There is something calming in reflecting on this unity within diversity. Beneath the wide array of tissues and organs, beneath the many shapes and behaviors of cells, lies a single origin, a single code. The skin cell, the neuron, the muscle fiber, the immune defender, all are siblings, descended from the same beginning, carrying the same instructions within. Their differences are not divisions but expressions of possibility, a quiet unfolding of potential into many forms. Differentiation is not only about development but also about renewal. Every day, cells in the body wear out and must be replaced. Stem cells and progenitor cells divide. This continuous renewal keeps tissues fresh, maintains balance, and ensures survival. Without differentiation, healing would not occur, growth would not proceed, and the body would remain static. With it, the body becomes dynamic, always changing, always adapting, yet always sustained. As we imagine differentiation, we might picture a single river branching into many streams. All the water comes from the same source, yet each stream follows a different path, irrigating fields, nourishing forests, or flowing quietly to the sea. The streams are different, but they are not separate, for they remain connected to the river. In the same way, differentiated cells are distinct in form and function, yet united in origin, all flowing from the same source of life. In the next part, we will move from differentiation to regeneration, exploring how tissues repair themselves after injury and how cells work together to restore what has been lost. By studying regeneration, we will see not only the power of renewal but also the limits and variations of healing, both within humans and across the wider living world. 19. Regeneration and repair. Life is not free from injury or wear. From the smallest cut on the skin to the unseen strain within tissues, the body is constantly challenged. Yet built into every living organism is the quiet capacity for renewal. When damage occurs, cells respond. They divide, they migrate, they differentiate, and they work together to repair what has been lost. This process, known as regeneration and repair, reveals the resilience of cellular life. It is a reminder that though fragility exists, so too does restoration, and that healing is as natural a rhythm as growth itself. In humans, regeneration is not absolute. We cannot regrow entire limbs as some salamanders can, nor can we replace all organs once they are damaged. But within the limits of our biology, regeneration is steady and persistent. Skin renews itself continuously, closing wounds with layers of fresh cells. Blood is replenished daily, with new red and white cells arising from stem cells in the bone marrow. The lining of the digestive tract, exposed constantly to friction and acids, is rebuilt every few days. These examples show that repair is not rare, but constant, woven into the very fabric of life. When an injury occurs, the body responds in stages. First comes recognition. Cells at the site of injury release signals, summoning immune cells and growth factors. These signals are like beacons, calling for assistance. Soon, specialized cells arrive, cleaning away debris and defending against infection. At the same time, blood vessels dilate, increasing the flow of oxygen and nutrients to the damaged area. This initial phase may be accompanied by inflammation, a natural part of repair that clears the path for healing. Next comes proliferation. In this stage, cells begin to divide and migrate. Stem cells and progenitor cells play a central role, producing the types of cells needed to rebuild tissue. In the skin, for example, basal cells divide and move upward, closing gaps in the protective barrier. In the liver, hepatocytes divide to replace lost tissue, demonstrating one of the most impressive regenerative capacities in the human body. In bone, specialized cells lay down new matrix, gradually knitting fractures together. Each tissue follows its own strategy, yet all share the same principle: renewal through cellular effort. Differentiation is essential during repair. Cells must not only multiply, but also take on the proper roles. In wound healing, fibroblasts differentiate into myofibroblasts, which contract to draw the edges of a wound together. In blood vessel repair, endothelial cells differentiate to form new capillaries, restoring circulation. This orchestration ensures that the rebuilt tissue is functional. Repair is not random but carefully guided, a quiet choreography of specialization. Sometimes, when damage is too severe or regeneration incomplete, repair results in scar tissue. Scar tissue is formed by connective cells laying down dense fibers of collagen. It restores strength and integrity, but it lacks the full function of the original tissue. Scarred skin may be less flexible, scarred muscle less elastic, yet even scar tissue is evidence of resilience, a compromise that preserves structure when perfect regeneration is not possible. The body prioritizes survival, ensuring continuity, even if some function is reduced. Across the natural world, regeneration varies widely. Salamanders can regrow entire limbs, complete with muscles, nerves, and bones. Certain fish can regenerate parts of their hearts, while flatworms can regenerate entire bodies from small fragments. Humans, by contrast, display more limited regeneration. Still, the principles remain shared. Stem cells, growth factors, and careful signaling guide the process. The differences lie not in the existence of regeneration, but in its extent. By studying other organisms, scientists glimpse the broader possibilities of cellular renewal, possibilities that hint at what life can achieve. Even within the human body, some tissues stand out for their regenerative capacity. The liver is perhaps the most remarkable. If a portion of it is removed, the remaining tissue can grow back, restoring mass and function. The skin, too, is endlessly renewing, maintaining its barrier through cycles of division and differentiation. Blood, with its rapid turnover, is constantly replenished. These tissues remind us that repair is not rare but ongoing, a silent process that unfolds throughout life. The nervous system shows the other side of this story. Neurons in the brain and spinal cord regenerate poorly, if at all. When injured, they are more likely to be lost permanently, and the connections they form may not be replaced. Yet even here, there is some capacity for adaptation. Other neurons may strengthen their connections, forming new pathways to compensate for loss. This plasticity is not true regeneration, but it allows for recovery in its own way, revealing the quiet determination of the body to endure. Regeneration and repair are also deeply tied to the concept of balance. The body must replace cells when needed, but not in excess. Too little repair leads to decline, Too much growth leads to imbalance. This is why repair is guided so carefully by signals and checkpoints, ensuring that the response is appropriate. The balance between renewal and restraint is as important as the act of regeneration itself. There is comfort in knowing that repair is always at work within us. Even as we go about daily life, unseen processes are mending small tears, replenishing lost cells, and restoring equilibrium. A scrape on the skin, a minor cut, or the everyday wear of tissues all set in motion quiet cascades of renewal. These processes seldom call attention to themselves, yet they protect the integrity of the body from moment to moment. To picture regeneration, one might imagine a quiet meadow after a storm. The grasses may be bent, the flowers bruised, the soil disturbed. Yet, in time, shoots rise again, flowers bloom, and the meadow regains its stillness. The traces of the storm may remain, perhaps in a scar on the landscape, but life continues, renewed and steady. In much the same way, tissues recover, carrying forward the resilience of life. In the next part, we will explore the immune system at the cellular level, looking at how specialized cells defend the body from harm. Through their vigilance and coordination, we will see how cells not only repair, but also protect, preserving balance in a world full of challenges. 20. The immune system. Within the body, there is a vast community of cells, each with its own task, each quietly sustaining life. Among them, some cells carry out a very particular role. They defend the body against threats. These defenders form the immune system, a coordinated network of cells and signals that recognizes danger, responds to it, and restores balance once the danger has passed. Unlike barriers such as the skin, which simply block what is outside, the immune system is active, alert, and adaptive. It remembers past encounters, it learns from experience, and it tailors its responses with precision. To reflect on the immune system is to see the body not only as a structure of tissues, but as a vigilant community that protects itself with order and care. At its foundation, the immune system relies on two main strategies: innate and adaptive immunity. The innate system is ancient and immediate, a first line of defense that responds quickly to general patterns of danger. It includes barriers like the skin and mucous membranes, but also cellular guardians such as macrophages and neutrophils. These cells patrol tissues, engulfing invaders and clearing debris. Their responses are fast, but not highly specific. They act like sentinels, protecting broadly against what does not belong. The adaptive system, by contrast, is slower to respond at first but far more specialized. It depends on lymphocytes, B cells, and T cells that recognize particular features of pathogens. Once activated, they mount targeted responses, producing antibodies or directly attacking infected cells. More remarkably, they remember. After an infection has passed, memory cells remain, ready to respond more quickly if the same threat returns. This memory is the basis of long-term protection, a quiet archive of encounters stored within the immune system. Among the many players of immunity, macrophages stand out for their versatility. These large cells move through tissues like caretakers, engulfing bacteria, clearing dead cells, and releasing signals that call other defenders. Neutrophils, smaller and more numerous, rush quickly to sites of infection, overwhelming invaders with sheer numbers. Natural killer cells add another layer, identifying and destroying cells that are abnormal, such as those infected with viruses. Each type has its specialty, yet all contribute to the shared purpose of protection. B cells and T cells bring sophistication to the system. B cells, when activated, produce antibodies, molecules that circulate in the blood and tissues, binding specifically to antigens on pathogens. These antibodies mark invaders for destruction, neutralize toxins, or prevent viruses from entering cells. T cells, meanwhile, come in several varieties. Helper T cells coordinate immune responses by releasing signals that guide other cells. Cytotoxic T cells directly attack infected or abnormal cells, ensuring they do not spread harm. Regulatory T cells temper the response once the danger has passed, preventing excessive damage. Together, these lymphocytes provide precision, adaptability, and balance. The coordination of immunity depends heavily on signaling. Cells of the immune system release small proteins called cytokines, which act as messages. These signals attract other cells, increase activity, or calm the system when the task is complete. The constant exchange of cytokines ensures that the response is proportional, strong enough to eliminate the threat, but restrained enough to avoid harming healthy tissues. This balance is essential, for immunity must protect without overwhelming. The immune system also depends on learning. During development, lymphocytes are generated with a wide range of receptors, each capable of recognizing different patterns. Most will never encounter their specific match, but a few will, and when they do, they expand and strengthen. In this way, the immune system prepares for countless possibilities, holding within it the capacity to face dangers it has never seen. It is a remarkable example of biological foresight, a quiet readiness for what may come. In addition to defense, the immune system contributes to repair. After clearing infection, immune cells release signals that encourage healing. They stimulate the growth of new blood vessels, the deposition of fresh connective tissue, and the restoration of normal function. In this way, immunity is not only about fighting but also about rebuilding, ensuring that tissues return to balance once disturbance has passed. There is also an element of tolerance within immunity. The body must distinguish between what is self and what is not. To achieve this, developing lymphocytes are tested, and those that react strongly against the body's own tissues are removed. This process reduces the risk of attacking the self, though it is not perfect. The careful balance between recognition and tolerance illustrates the subtlety of immunity. It is a system that defends fiercely, yet must restrain itself from misdirected aggression. In everyday life, we rarely notice the constant work of the immune system. Each breath of air brings microbes into the lungs. Each meal carries potential invaders. Each cut opens a path for bacteria. Yet, without effort on our part, the immune system responds. Cells patrol, signals are exchanged, defenses are mounted, and balance is restored. Only in illness do we sometimes glimpse its activity, through fever, inflammation, or fatigue. These are signs not only of stress, but of a system at work, a reminder of the hidden guardians within us. It is soothing to reflect on the immune system not as a battlefield, but as a community of caretakers. Macrophages cleaning, lymphocytes remembering, cytokines guiding, all are participants in a quiet vigilance. Their work is not frantic but steady, not chaotic but organized. They are always present, always prepared, ensuring that the larger organism can continue in peace. As we imagine this system, We might picture a village protected by gentle watchkeepers. They walk the borders, guard the gates, and respond to disturbances with care. When danger passes, they return to stillness, allowing the village to thrive in calm. The immune system is such a presence within us, maintaining security so that life may unfold without constant awareness of threats. In the next part, we will explore how cells connect with one another to form the nervous system, the vast network that not only responds to the world, but also creates sensation, thought, and awareness. By looking at cellular contributions to the nervous system, we will see how communication reaches its most intricate form, weaving perception and consciousness from countless tiny signals.